So I'll just see the questions pop up in Q&A, and we might have to remind people in the chat box to put in questions in the Q&A, but except for that, as they pop up, we'll just speak them out. Got it.
Yeah,
okay.
I'll make sure, I'll monitor the chat for that.
Perfect. Okay, sounds good.
Okay, perfect. Let's start the webinar, I guess. Yeah, because we haven't started the webinar. You sent it, but nobody will actually see it. Okay, we're going live in 3, 2, 1.
Okay, hello everyone. I'm the host for this session along with Taha Murshid-Zazeh, and we have 3 very exciting speakers lined up, and they're going to use, it's truly multimodal, MEG, EEG, fMRI to study the human brain. It's a 12-minute presentation with 3 minutes for Q&A, and you can put your questions in the Q&A box. The first speaker is Luke Tate. He is a research associate at the Cardiff University Brain Research Center, and he's going to talk about his work on optimal source reconstruction of resting MEG of the human brain.
Great, thank you. So I'll just share my screen with you. Great, so hopefully you should be able to see my screen. Yeah. Perfect. So as mentioned, I'm going to talk to you about some work I've been doing looking at source reconstructing resting state MEG data. So just to give you a quick idea, the idea of source reconstruction is given some MEG or EEG activity recorded outside of the scalp, we want to estimate the brain activity that generates this data. Now, in the literature, there are many, many different methods for source reconstructing EEG or MEG data. So we wanted to ask, what is the best way to do this? Specifically for resting state MEG or single trial data, which is distributed across the cortex, low signal to noise ratio, compared to, for example, evoked potentials. So to address this problem, we took 11 participants with 30 seconds epoch of pre-processed, artifact-free resting state MEG. So we used a 274 channel CTS scanner for each participant. And the forward model, we use a single shell head model derived from a T1 weighted MRI. We studied 6 commonly used inverse algorithms. So there were 2 beamformers, the LCMV and its depth weighted counterpart, that here we've called weighted LCMV, and then 4 least squares minimum norm type estimates that are common in the literature. So in order to compare the algorithms and quantify which are the best algorithms, or which perform well in different aspects of MEG data, we wanted to look at spatial and temporal quantifications. So for the temporal aspect of the data, we wanted to say, can the algorithm explain the temporal fluctuations due to brain activity? So we took our resting state MEG, we left out a subset of electrodes. So here I've only shown 1. In fact, it was between 10 and 11, and we did 27-fold cross-validation. So we left out a subset of electrodes, and we source reconstructed the remaining electrodes. Then, given our source solution, we forward mapped back to these left out test sensors. And we calculated the temporal variance explained, so the correlation squared between the predicted activity at those sensors and the real activity that was recorded at those sensors. So there's a slight downside to this method in that MEG data resting state MEG data has low signal to noise ratio. So explaining all of the variance means that you're also explaining the noise in the data. So in order to try and penalize noise and account for this bias, what we did was we also calculated this metric in empty room data, where there is no brain activity at all. And then we divided by the variance explained in that. So now we've got a ratio of with a brain versus without a brain, how much variance do we explain? So that is to explain temporal fluctuations in the data. What about the spatial aspects? To do this, we use something called the resolution matrix. Now, I won't go into the details here, but this is a theoretical relationship between the estimated source activity and the true source activity that we can't know. And from this matrix, we can get something called the point spread function. But that says, if I've got activity at a dipole, here marked with a diamond, where does the estimated activity from this dipole, sort of where do we estimate this activity is? And so we can look at something called the localization error, which is the distance between the dipole and its peak of the estimated activity. Or we can look at the spatial extent of this. How far does it spread out? So here we've called it spatial extent of point spread. So this says how far does leakage come from the dipole? We can also look at something called the crosstalk function that basically says, given an estimate at a dipole, how far away is activity influencing this estimate? So we can look at the spatial extent of the crosstalk function to calculate how far away is leakage into the dipole. So we've got out from the dipole and into the dipole. So these are our metrics to look at the spatial aspects and quantify how good spatial reconstruction is. And finally, we know that resting data is low signal to noise ratio. We can't average over many trials to reduce noise. So we wanted to see, is the algorithm strongly biased by noise? We can't reduce the amount of noise very easily, but we can increase it. So to quantify this, what we did is we first of all source reconstructed our resting state meg, and then we added some sensor noise and source reconstructed this. And then we performed a spatiotemporal correlation between these two solutions to see how much the solution changes with the addition of some noise. So first of all, we said the temporal fluctuations, the R-squared analysis. And what we found was that the beamformers greatly outperformed the least squares minimum norm type estimates. And this was actually primarily due to the fact that they explained very, very little of the empty room data. They were, whereas the others slightly overfit to the noise. How far away was the activity misplaced, the localization error? Well, we found that the LCMV beamformer had the highest localization error, whereas, as theoretically expected, S Loretta and E Loretta had 0 localization errors. The two spatial extent of leakage measures gave very similar results, actually. So I'll present these together just as a general measure of leakage. As you can see, the beamformers performed worst of the group. They had the highest leakage, whereas the weighted minimum norm estimate had the lowest leakage. And finally, with the noise analysis, with the adding the noise, so on the x-axis here, we've got the amount, the variance of the noise as a ratio of the variance of the data. And you can see for different amounts of noise, the MNE and weighted MNE solutions, the correlation drops a lot faster than the other solutions. And this just sort of quantifies it as the sort of the slope of this drop off. So you can see MNE and weighted MNE drop off a lot quicker. So that's a lot of information in a short amount of time. So I thought it'd be handy if I summarize this all in a big table, summarize these results and give some conclusions. So first observation was that the depth weighted metrics, the weighted LCMV and the weighted MNE, always performed better than or equal to the unweighted counterparts, LCMV or MNE, in all of the metrics that we looked at. So based on our results, we should prefer the depth weighted metrics. This is kind of expected. The depth weighted is supposed to improve the result. So I've left out the unweighted metrics just to clean up the table a little bit here and just looked at the remaining 4. So I would say if the data, we know it's particularly noisy, we know it's noisier than, noisier than the typical analysis, I would recommend using a beamformer. They explain very little of the empty room data and were very robust to noise. So the extra noise is less likely to influence your results. However, this does come at the cost of having high localization error and high leakage. If you prefer to use a least squared minimum norm method over beamforming for whatever reason, and the data is still very noisy, you've got your noisy data, then S Loretta was the way to go. It had, out of all the metrics actually, the highest robustness to the noise. but it did drop in the variance explained. So again, you win some, you lose some there. If having high resolution is your top priority, if you really want to minimize leakage and are happy to take in the localization error, the less robustness to noise, bias to noise, a loss of variance explained, then by all means use the weighted minimum norm estimate, but it comes with many downsides. And finally, if you don't think the data is particularly noisy, if you think it's just fairly standard resting state data, if you don't have particularly strong requirements that it has to be minimize leakage as much as possible, I would say of the group, eLoretta performed generally the most well across the board. It was very rarely the best, but it performed reasonably well in most metrics. So I think eLoretta is a strong choice here. Now, in resting state analysis, typically, actually, we would parcellate the data into regions of interest as opposed to just perform the analysis on the level of voxels. So I won't go into details here just because of time constraints. But if you look at the preprint presented given in the chat, we reduced the Human Connectome Project's multimodal parcellation down to 230 ROIs specifically targeted towards use with MEG. So this atlas is freely available for download at my GitHub page given down here. We also validated this atlas in a larger sample of 31 participants with 2 scans per participant. So this isn't just the 11 participants that we used in this data, in this analysis. So we modified our metrics slightly to be more suitable for ROI level analysis and reevaluated these metrics. So I'll just show you again another summary table here. So the first thing that I really noticed was the fractional localization error. So this was, instead of just saying what is the distance between the dipole and the peak of the estimated activity, I said what fraction of dipoles have a peak of activity outside of their ROI. It doesn't matter if the peak of the activity is inside the ROI but a few millimeters away, but it does matter if we're putting it in the wrong ROI. And both weighted LCMV and weighted MNE had over 50%, in fact, over 65% really, of dipoles misplaced into the wrong ROI. So I should say here, this is a very high resolution atlas. If we use, say, the AAL atlas, which only has 78 regions, this might be less of a problem. But for this high resolution atlas, it really means that this is quite a problem using these metrics. So given that we don't want to use either of these metrics for this atlas, we want to use either eLoretta or sLoretta. And actually, if we just look objectively at the solutions, eLoretta outperformed sLoretta. There's not much between them, but eLoretta outperformed them both slightly. So I would recommend the use of eLoretta for high resolution atlases. So that's all my slides. Are there any questions?
Thank you. And it was perfectly on time. Let's see if we have questions from the audience. I saw someone had raised their hand in the audience, but I don't see a question in the Q&A box. Did you have a question that you wanted to type in?
Yes, please send your question in the Q&A section, and we will address it live. If you would like to, after you put it through the Q&A, we can bring you forward to discuss the question further in depth with the speakers as well. Oh yeah, Marco, you can go with the question as well. You can unmute and ask your question.
Okay.
Since there's a question.
First of all, thank you. Just a great presentation.
Thank you.
I was wondering, how do you think this would compare with task data? Is there any reason for it not to be the same type of results?
So, yeah, I mean, the main reason that I don't want to strongly say that this would also apply to task data is because some of the measures rely strongly on the covariance matrix, so particularly the beamformers rely on the covariance matrix of the data. And if you are averaging over many trials, this is going to greatly change the covariance matrix. Also, these are all regularized solutions, so changing the SNR might change the results as well. You can absolutely apply these analysis methods to the resting state data. And at the GitHub page that I showed on my other Atlas slide, at this GitHub page, I've also provided codes to do all these analysis on task-related data. So it's absolutely something you can do, but I don't want to say now that it would work for task data just because we haven't done it. You know?
Okay. Just another question. The decrease on the variance when you move to the Atlas version on the beamformer method, was that expected?
So the reason for this is, in the actual paper, we show the R-squared and the empty room data separately. In the Atlas version, what we found was that all of the metrics throughout only explained like 10 to the negative 4 variance once we parcellated. So I suspect we used the first principal component of all dipoles within an ROI to parcellate. I suspect by rejecting all the other principal components, we're sort of rejecting noise. So in this case, we actually relied just on the variance explained, which was lower for the beamformers in the original solution. It's just that in the voxel-wise data, the least squares minimum norm explained noise as well, whereas that doesn't happen in the parcellated solutions.
Okay, maybe just one last question. How do you think this will generalize to EEG data? Would it be the same? Do you think it?
I think so. There has been some people looking at this with the resolution metrics. It's quite dependent on the EEG, the density of the EEG. I think if we had very, if you had very high density EEG, the results would be fairly consistent. Of course, you've got the difference in the radial versus tangential, which means that, so I haven't included here, but we did plot, because each of these metrics are per dipole, what we showed here is averaged over the whole brain. We did show plots, and that I think the spatial distribution would be different, but I think it was largely.
Sincere apologies for interruption, because we are running more than a minute over time. We might hit the spot where our webinar will be just ended and we'll be kicked out. So yeah, let's just move on.
No problem.
Keep the Q&A going in the chat. Just keep talking about it. This is interesting. I'm learning stuff from this as well.
Thank you.
Right, let's transition to the next speaker. It's Bei Huang. He is a research fellow at the Singapore Institute of Clinical Sciences, and he's going to talk about correcting for superficial bias in gradient echo fMRI.
All right. Okay, hello everyone. My name is Huang Pei, and I'm actually presenting some of my previous work done at the MRC Cognition and Brain Sciences Unit at the University of Cambridge. So I'll be talking about correcting superficial bias in 70 gradient echo fMRI, and I'll be approaching this using both computational simulations and actual fMRI results. So gradient echo fMRI is a very popular sequence for studies at 70 because firstly, it's very simple to apply and it's very easy to implement in scanners as compared to some of the newer sequences such as ASL or Bustle. And it also has the advantage of having some of the highest signal to noise ratio. And so like there's a figure here which shows the comparison between the various methods that can be used for fMRI at 70. But of course, we must highlight that gradient echo has a big drawback, which is the presence of superficial bias. And this is where larger signal changes are observed in the superficial layers as compared to the deep layers. So the problem with superficial bias is very well established in literature, as you can see from all these figures. And all of them show the near identical trend of increasing signal towards superficial layers across all their different paradigms. And this also observed to affect both raw responses as well as experimental effects, such as attentional modulation, as seen from these two 2020 papers. Another interesting thing to highlight here is that there's also another trend present here, which is there's increasing variance as we go towards the superficial layers. And this shows that there's likely some correlation between increasing signal and increasing noise. And this is actually something that some researchers have tried to use to correct for superficial bias, as I'll show later. Okay. So to solve the problem of superficial bias, multiple different methods have been utilized in the literature, and people have also attempted to model the bias. So the below figure here, this figure, is taken from Haber's blog, and it beautifully illustrates the three main methods: linear offset, multiplicative, and leakage models that are being explored currently, as well as the proposed methods of correction for the respective models. So with this understanding of the background, we set forth our goals, which is to build a computational model to generate simulated fMRI data. We can then test out various correction methods on this data and evaluate how good each method is. Finally, we can then validate the results from our model on real fMRI data. So to give a brief illustration of paradigm, I won't go into too much detail because of time constraints, but I just want to highlight that we have two task conditions, which is task D plus and task D minus, and they differ in that there's a distractor present or absent, while the participant is attending to the diagonal of interest. And if you're interested in a paradigm, our paper, actually, we have published a paper on this data already, and it's described in this paper, link below. Okay, so for our computational model, we decided to use the multiplicative model of superficial bias. We simulated a time course of 2,500 voxels, which is roughly the size of the ROI in our real data. And we started from a neural level. So the voxel response for each stimuli is obtained by taking the product of the firing rate, the neural density, and the attentional modulation. The firing rate is simply 1 when the stimulus is present and 0 when absent, while the neural density for each voxel is randomly sampled from a half normal distribution. The variance of the distribution is adjusted to capture the global preference of the ROI. So for example, if we were to simulate the FFA, we will sample from a half normal distribution with higher variance for phase responsive neuronal density. The attentional modulation is set as 1 for passive viewing and greater than 1 for attending. And the total voxel response is then obtained by summing across all stimuli conditions. Then we generated the voxel time course by multiplying the voxel response with the design matrix of our experiment. Lastly, we added in physiological and thermal noise, as well as the multiplicative superficial bias to obtain the overall measured time course, which is what we get from our fMRI data. So the parameters for our simulation were chosen to best fit our fMRI dataset. As you can see from the figures here, the mean and the standard deviation of the t-statistic of our simulation closely match that of our real data. And we generate a scatter plot of the voxel responses for the simulated data and the real data. You can see that they are in good agreement as well. We validated 6 different correction methods in our simulation, and each method was iterated over 10,000 times to generate an average result. And if you're interested in our code or the actual simulation results, our GitHub page is linked here. Okay, so with our simulation setup, we chose these 6 methods to evaluate. Of this, some have been used in the past, while some are more novel. For example, Deming regression is a method proposed by us as an improvement to the ROI ratio metric previously used. So we can broadly classify these metrics into 3 groups: the ratio metrics, z-scoring, and multi-voxel pattern analysis methods. So the 6 underlying methods are what was used in our simulation. So ratio metrics are a very intuitive approach if we expect the bias to follow a multiplicative model. By taking a ratio of the 2 signal changes, we can remove the effect of the bias L and recover the ratio of the underlying response as seen from this equation. However, because we have a large number of voxels, there are multiple ways to take the ratio. So firstly, the simplest way is to calculate a ratio for each individual voxel and average across all voxels. However, this method is prone to be distorted by extreme values, especially for voxels when task D minus contrast is close to 0. So this whole ratio will go to become a very large number. One way around this, which is the ROI ratio, is to take a sum or average across all voxels first, and then taking the ratio of the 2 metrics. In this way, you can regularize it to become more controlled, but it comes at a cost because this becomes a univariate metric, and you can discarding potentially important regional pattern information. Lastly, Deming regression, which is proposed by us, is by simply obtaining by regressing one condition against the other and then taking a gradient. So we chose to use Deming regression instead of ordinary least squares because Deming regression accounts for the error in both variables. An alternative approach, which has been previously used by Lawrence et al. And others, was to normalize the original time course using z-scoring before fitting the GLM. So this is what I was talking about when they're trying to use the noise to normalize the signal. And so that's the idea that they're trying to implement here. And lastly, the last class of correction methods is the MVPA methods, where we evaluate the ability of the classifier to classify the attended category using voxel information. So this approach similarly relies on the observation that both noise and the signal of interest scales with superficial bias. And here we evaluated two methods, a classical support vectors machine SVM and a linear discriminant contrast method. So these two diagrams illustrate the differences between the two methods. This is a simple two voxel example. And if you're interested in reading more about the comparison between the two methods, we have a paper link below that goes over it. So we first simulated on the most basic case, which is constant attentional modulation across layers. So on the far left, you can see that you have a very nice flat attentional profile that is fed into the model. And the measured response shows a clear increasing trend towards the superficial layers, showing that our model is generating data with the expected profile. For the ratio metrics, you see that both Deming regression and ROI ratio are able to accurately capture the flat attentional profile, while voxel ratio shows high instability, similar to our expectations. Z-scoring, SVM classification, and LDC all show a strong superficial profile, superficial bias, and thus are probably unsuitable for NS for usage. So next, we simulated a V-shaped attentional profile, which is what we might expect based on previous neuroanatomical data. Similar to what we observed with constant attentional modulation, both Deming regression and ROI ratio is able to recover the underlying attentional profile, whereas voxel ratio is highly unstable. And you can see that it's unsuitable because the noise will definitely mask any true attentional profile that's underlying. LDC, SVM, and Z-scoring all show a V-shape, all show somewhat of a V-shaped profile, but there's still a substantial amount of superficial bias present, so it's likely unsuitable for this purpose. Lastly, to highlight the difference between univariate and multivariate approaches, we repeated the same simulation, but we removed the global preference, which means what we did was we sampled both neural densities from the same half normal distribution. So you can see that from the measured response that there isn't a regional mean contrast left over. So thus, because of this lack of regional mean contrast, ROI ratio, which is a univariate method, becomes highly unstable as well. Whereas Deming regression, which is more multivariate, will be able to still capture the underlying attentional profile and still stable. So similarly, SVM classification and LDC, which are both multivariate methods, are also able to capture the V-shaped profile. But again, there's still an element of laminar bias present, which is why they're unsuitable. So we now applied the 6 correction methods to our data, our real fMRI data set. You can see that our raw data has nice laminar bias, but when we apply like Deming regression or ROI ratio, they both show very nice horizontal profiles. And whereas like voxel ratio is highly unstable, you can see from the insert, it's extremely unstable and definitely should not be used to analyze the laminar data. Z-scoring, SVM classification, and LDC all show a somewhat superficial bias. And this is what we're trying to highlight, is that it's important to correct for superficial bias, because if you were to look at one of these 3 metrics, or like even all of these 3, you might draw an erroneous conclusion that there's stronger signal in the superficial layers. Whereas like based on our simulation and our data that I showed previously, it's likely that the correct conclusion from this set of data is that it's likely that there's like a flat attentional profile as shown by Deming regression or ROI ratio. And so in conclusion, I would like to stress the importance of correcting for superficial bias in gradient echo fMRI, because it's very easy to draw wrong conclusions based on the superficial bias. And so using our results, we recommend using ROI ratio or Deming regression depending on whether there is a mean global preference or no global preference. Deming regression also has an added advantage in that it's a multi-voxel approach, so it might be preferable if there are other voxel pattern data that can be exploited. However, it's important to note that our results is only simulating a multiplicative bias model. And while we have some validation from real fMRI data, it's possible that alternative models, such as leakage model, might give different conclusions. And this is something that we would like to work on improving with our future simulations. All right, so I would like to thank my supervisors, collaborators, and everyone else who helped out with our work. Yeah, and thank you. So this is the link to our code and my email if you have any questions.
Thank you. That was a very nice and concise presentation. And the link is also in the chat box for the attendees. Any questions? And the panelists are welcome to ask questions as well. Yeah, I had a quick question more in terms of, I know now you've transitioned to a new lab. Are there other research questions and data sets that you're going to apply these algorithms to, these methods to? And do you think the results or the outcomes might be different depending on the area or the task?
So I think that this is actually, because this simulation is a very general simulation, I think that it actually applies to almost all gradient echo data. And it was like, if you go back to like my introduction, I think, if I show here, you can see that because there's a wide range of paradigms have been explored at 7T, and all of them still show very similar profile. So it's likely that this is something that is constant across all regions.
Thank you. Any other questions? Last chance, one more question. All right, thank you. And we'll transition to our third speaker. I have information there in the chat box about the third presentation, Dr. Marco Simoi, and he's going to talk about EEG fingerprinting.
Okay, hello everyone. My name is Marco and I'm presenting the FitPool method, a new EEG fingerprinting method for transferring fMRI-based neurofeedback interventions to EEG setups. This work has been conducted in University of Coimbra in Portugal. Okay, so basically what is neurofeedback? We just take the signal measurement from a specific brain process of a subject and we feed that information back to the participant so it tries to self-regulate its own brain activity. This process can usually be done with a neuroimaging method. So here we compare two neuroimaging modalities, the fMRI and the EEG. The fMRI-based neurofeedback, we have a very high spatial resolution, which gives us a good anatomical validity, but we have a low temporal resolution, and most of all, we have an inflexible setup that is very expensive. So when we are talking about a method that we want to use for intervention and for rehabilitation, this can get very expensive. However, on the EEG side, we have a very low spatial resolution, which gives us poor anatomical validity. But in contrast, we have a high temporal resolution, and it provides us with a flexible setup, which is very inexpensive. And we can do remotely, we can go to the patient's homes and do the interventions there, which we cannot do with the fMRI. So this leads to two different characteristics of the EEG modalities led to different characteristics on the methods used for neurofeedback. On fMRI based, we have usually localized regions. We define a region of interest and we try to modulate the bold signal on that region a long time. However, in contrast, on the EEG neurofeedback approaches, we usually use more broad rhythmic modulations where participants try to modulate the frequency bands of the signal. The fMRI-based approach is much more recent. We're talking about 1-2 decades, while on the EEG base, we have it for the 50 to 70 years in the literature. However, the EEG setup provides still poor evidence of effect, while the fMRI-based approach start to show some promising results. However, due to the constraints I talked earlier, the number of sessions per study is much different. So on the fMRI-based approaches, we have 1 to 5 sessions at most on the literature, while on the EEG neurofeedback, we have usually from 30 to 50 sessions. It's fairly common to find. So our idea was to develop an fMRI-based neurofeedback intervention and try to transfer it to EEG. How did we aim to do this? We acquire simultaneously EEG and fMRI signal on the participants doing the fMRI neurofeedback intervention. With the data acquired simultaneously, we try to predict the bold activity on the ROI region based only on EEG data. And with that model, we then are able to use and do the same task, fMRI-based neurofeedback, but using an EEG setup outside of MRI. So this is the main idea. For that, we developed a neurofeedback protocol where we applied to 10 healthy participants. We localized the posterior superior temporal sulcus region with a stringent localizer. And we conducted 1 neurofeedback session of bold modulation on this region using imagery of facial expressions to increase the activity on that region. So with this, we aimed to provide the feasibility, and we published this paper on the Neuroscience Journal. With that data, we looked in the literature and we found that there is this group on Tel Aviv which developed this method called the electrical fingerprint, where they use the power on different frequency bands on the previous 12 seconds of the signal and use that to predict the activity the bold activity on the ROI. With this method, they have already published several papers where they use the EEG method transferred from the fMRI. So they are using it clinically very often. So we were excited because it seemed to be a good method for us to use. We then thought on improving the method. So we thought, instead of only using the power, try other features from the linear and also the nonlinear feature space. So we tested different frequency bands. We do it on the scalp signal and also on the source signal. We reconstructed the time courses on the source space. And then we used the same method they did for dealing with the hemodynamic delay on the fMRI, the delays method, but also compared it with the HRF, the commonly used HRF convolution. And also we tried to do a multiple HRF convolution where we defined HRF's hemodynamic response functions peaking at different latencies and convolved the signal with all of them and then used feature selection. For a classifier, we used the random forest. This is a short pipeline of the approach. So we mapped the region of interest. We extracted the time course of the bold signal from the fMRI data on that ROI. And then this was our target signal. And from the EEG data, we performed feature extraction. On the scalp, we just used the multiple HRF convolution, but also than that, we conducted source imaging, do some spatial filtering techniques, and then do the multiple HRF convolution. So our results, we see that the FP methods showed a very low correlation, only about 20%. And with our method, we were able to increase about 30 percentage points the correlation achieved between the predicted bold from the EEG data and the real bold from the ROI. Also then that we can see that we increase the anatomical validity of the reconstructed signal, because when we do this source localization method, we see that with the EFP method, we did not overlap a lot with the bold signal, which we see in blue. While on our method, we can see a nice overlap. We can see in green between the source signal predicted by our method and the original localizer. And then we look to the importance of the different features. And we see that the nonlinear features were having the most contribution to the method, and also that the theta and alpha frequency bands were the ones most used. So in conclusion, we verified that nonlinear features might be important to capture bold relevant dynamics, which improved the reconstruction accuracy of the model. Also, the convolution with multiple HRS functions work better than the delays method or the simple HRF convolution. And we verified that this increased reconstruction is needed to achieve a better anatomic validity of the methods. There are still some work to do. We need to assess the generalization of these models. So how well these generalize between subjects, and even for the same subject for different sessions, because we see a lot of variability both between subjects and also intra-subject. And we can see that in both modalities, both in the fMRI and on the EEG. So this is a big challenge that we need multi-session EEG fMRI data sets, which are very difficult to find on the literature. Also, we wanted to check if this method can be applied to other neurofeedback tasks in different target regions, because this region is really challenging because it's small and it's in a sulcus. And also there is this big question, then how valid must how accurate must this reconstruction be so it is valid for transfer? Because we see that the method that gives us 20% correlation is already being used and validated in clinical trials. So we have this question to know how much is needed to verify and apply the method. So this was my presentation, and I am open for questions.
Thank you very much. That was a nice presentation. Hopefully this work brings us one step closer to translation of neurofeedback. We do have one question already in the chat box. Shagun is asking, could you tell us about bold activity was predicted for how long in terms of future duration and how many scans?
Okay, so we have, it was only one session, so one scan, but we did it on 3 runs, 3 different runs. And what we did was we predicted each time point, each TR of the bold signal using the previously 12 seconds of the EEG. So we have 12 seconds in the past to predict this point. Because of the hemodynamic delay. What we did is we convolved this with different HRF responses, and then we have this signal on these 12 seconds before. I'm not sure I responded to what Sean was asking, but he may confirm.
Sure. Any other questions? There's one more from Priyanka in the audience. Did the EEG have artifacts because of the simultaneous MRI scanning? And is the model applicable outside the MRI scanner?
Okay, so yeah, this is one of the big challenges of doing EEG fMRI, is the EEG contamination with the gradient artifacts, artifact. So what we did was we conducted a pre-processing, a standard pre-processing method. We believe the data were correctly cleaned, so we are free of these artifacts. However, of course, this affects the quality of the EEG, but it's the, well, I think it's the standard EEG fMRI procedure. So with these signals cleaned from artifacts, we were then trained the models. I believe the model is applicable outside of the MR scanner. I think it does not depend at all of the MR scanner. We need the MR scanner to validate the model. But if we could train the model using, imagine different conditions, so upregulation, downregulation, and try to predict just these two states instead of a continuous prediction, what we are doing is a regression, so we can try to correlate the signal. But if we can imagine passing this to binary classification or multi-class classification, we can do it outside the scanner.
Okay, thank you. So we're almost done. We have time for one more question. If you have questions for the other panelists as well, Dr. Tate or Dr. Pei. So just a brief pause in case any more questions. Yeah, I'm just curious for Marco, the last talk, do you have plans for a specific neurofeedback application that you're targeting?
Well, what we did here was we did a clinical trial, an intervention on autism spectrum disorder using this region, the PSTS region. And so what we saw some interesting results on the fMRI, but we only did like 5 sessions. So we wanted to see if we can transfer this method to the EEG, and we do it more intensively, we can achieve a better result. So that was the main driver for this work. But I think we can do it on several different applications, like motor rehabilitation, for instance, that is actually easier to perform.
Got it.
So that would be interesting.
Sounds good. Thank you. Certainly, I'd be interested in the motor rehab applications for sure. Again, thank you for to all the speakers and to Taha for helping with moderating this session, and have a good rest of the day.
Thank you.
Thank you.
Thanks, everybody. Have a good day or night.
This marker selectively overlaps in the cortex with these somatostatin NNOS cells. And so that was the point where we started this project at. I took several different routes to try and understand these cells. I examined the outputs of these cells and the inputs of these cells using various techniques. And I also looked at the function of these cells using optogenetics combined with high density electrophysiology. And if anybody has any one of these that they'd particularly like to start with, I can begin there. If not, I can just start from the beginning. I can start over here if Prezi works with me. Okay, so I first began looking at the outputs of these cells. I specifically targeted these cells with the fluorophore using a specific intersect genetic virus in order to express an EYFP fluorophore in these cells. I injected this virus in the visual cortex and then looked across the brain. I saw that these cells had projections, which you can see here, as far as 1.5 millimeters away on the same side. And then very interestingly, more than 2 millimeters away from that injection site, I also saw projections on the contralateral cortex. And this is very likely important for understanding the function of these cells. I next looked at the input of 2 somatostatin NNOS cells using monosynaptic rabies tracing. This allows me to specifically label the cell's 1 synapse presynaptic to these cells with eGFP. I'm tracing off of, or I'm looking at inputs to the cells in yellow in these images. Presynaptic cells are in green. We can see that there are both excitatory and inhibitory neurons that are presynaptic to these cells. The excitatory neurons have these apical dendrites. Among the inhibitory neurons are these putative neurogliaform cells that can be identified based on their morphology. Outside of the cortical input, there's also input from visual thalamus, specifically the LGN. Something that was particularly interesting about the cortical input to these cells is that it seems to be very strictly defined to primary visual cortex, with sharp drop-offs in labeled cells at the border of this area. The next thing I did was try to understand these cells using this high-density electrophysiology with silicon probes in combination with optogenetics. Once again, I specifically targeted these somatostatin NNA cells with a channel rhodopsin, and then specifically chose the type of optogenetic stimulation to do in order to minimize some of the caveats associated with optogenetics. We think that these cells are involved in generating oscillations associated with changes in arousal. And in particular, when an animal goes to an unaroused quiet state, there are increases in low frequency activity and decreases in gamma power. And with various different frequencies of sinusoidal optogenetic stimulation, each of these seems to be pointing to an increase in low frequency power and a decrease in gamma power, very similar to this unaroused state. To show you that this isn't just a fluke, we also used a white noise stimulation and looked at how it affected different frequencies in the local field potential, and once again saw that there was preferentially induced low frequency oscillations. We have in addition, we have several lines of evidence that make us think that this is something that's a little bit more physiological. I picked out one here. I can talk about some other ones in the bonus slides if we get there. But with this optogenetic stimulation, we're able to evoke something that looks a lot like the canonical downstate of the cortex that's usually observed during slow wave sleep. During optogenetic stimulation, we can evoke these events that have a positive deflection in the LFP, a reduction in gamma power, and a pause in multi-unit firing. There's a question in the chat: where were you recording with the probes, and what was the duration of the sinusoidal simulation? So I was recording in V1, primary visual cortex, and the duration of the sinusoidal stimulation was about 10 seconds with a 1 second break in between, and then repeating that many times.
There's also a question, Jacob, in the Q&A, if you want to take that as well.
Okay.
So with respect to these cells' contributions to slow waves during the sleep, as well as their long-range connectivity, could you give us some insight on how these cells might contribute to local sleep phenomenon?
Yeah, that's actually something that we've been thinking about, because there's some of these results where if you use a brain area particularly intensely during the day, you might see more intense slow waves in this area when the animal is sleeping during the night. And I think that's definitely something that that we're interested in looking at a little bit later, because as until now, there really hasn't been a good way to connect these network level phenomena to any sort of circuit level implementation, which is definitely an area where these cells could be important.
There's another question from Eric. Sorry, the previous question was from Mohammed. This question from Eric is, could you comment on the role of somatostatin versus parvalbumin in the initiation of slow waves?
Yeah, so there's been some work previously on different subtypes of inhibitory neurons and their role in generating slow waves. A lot of this work has been done in either slice preps or during anesthesia, showing that there are roles for these different subtypes. A lot of this I find a little bit tough to interpret based on it being either performed during anesthesia or in a slice, and I think that there might be different circuit mechanisms that are going on based on these changes. One of the real benefits I think of this work is that it's done in awake animals that are undergoing natural transitions from sleep to wake. Yeah, if there's something else that you were looking for, I can try to talk a little bit more on this. Does that answer the question?
We'll see if we get a follow-up question. There's a follow-up question from Mohammed, and he asks, but how are these neurons located within the brain in terms of regions, networks, and connectivity?
Yeah, so these cells, they're located all over the brain. The exact connectivity is a little bit unknown right now. That's sort of one of the things that I'm trying to answer. We have sort of these initial answers. They might project long range. They project across into contralateral cortex, but we're really trying to explore this in a much more fine-grained way. I've been labeling these cells and trying to clear whole brains so that we can do light sheet imaging and reconstruct the arbor of whole single cells, which I think will really give us a lot better answer as to what these could be doing.
Okay, can I ask a follow-up question maybe as well? So you mentioned in the labeling that you have both inhibitory and excitatory cells that get labeled with this somatostatin. Mm-hmm. And I was wondering whether in the EFIS you have any ways of dissociating which cell type leads to the particular effects you see.
Yeah, I think what we would really love to have is we would love to be recording from a lot of cells, be doing this optogenetic stimulation, and then be able to optogenetically identify these somatostatin nNOS cells in our extracellular recordings. Every single one of these recordings I try to do that, but so far we haven't found any. They're an extremely sparse population, so it would actually, like, one cell I think would be a real accomplishment with this. But I think it would be extremely interesting. But something that I'm not going to talk about here is we're also trying to do calcium imaging simultaneously with recording local field potentials, in which case we could specifically identify the somatostatin nNOS cells and correlate their activity to the activity of other cells that we're imaging at the same time, as well as to oscillations that we observe in EEG or ECOG or however we choose to record it.
Very cool. Are there any more questions? Any last questions for Jacob? One more.
Are these cells sensitive to anesthesia? That's an interesting question. I actually hadn't thought about that. We could definitely do it. So somatostatin cells in general, they become very silent during anesthesia, but these cells are likely different than your typical somatostatin expressing inhibitory neuron. But it definitely would be interesting to take a look at what their activity looks like under anesthesia.
Awesome, thank you so much. We have to move on in this for the sake of time. So next up is Fateme Nemantolazadeh. She's an MSc student in electrical engineering at Isfahan University. And her interests span the fields of statistical signal processing and computational neuroscience. And today she will tell us something about spatiotemporal dynamics of mesoscale activity in sensory cortex. So you want to share your screen? And
thank you, Dr. Moore. Yes, of course.
Great.
Okay, thank you. Hello, everyone. Here, the synchrony in spontaneous activity in somatosensory cortex has been analyzed. It is believed that spontaneous synchrony may carry information about the organization, circuitry, and function of neural networks. To test this, we used an array, a 10 by 10 array of microelectrodes to sample the local field potentials or LFP signals and spiking activity of clusters of neurons in the sensory cortex of anesthetized rats. And the field potentials acquired from sensory cortex were digitally filtered for the classical set of frequency bands, and then we quantified spatial dynamics of synchrony in LFPs in terms of phase amplitude and phase amplitude coupling at different frequency ranges. For phase coupling, we computed phase locking value across phases of two bandpass signals, and for amplitude and phase amplitude coupling, we computed a mutual information between envelopes or phase and envelope of pairs of frequency bands. You can see a co-occurrence of oscillations here, specifically in this case, I mean phase amplitude coupling. The lower gamma range and delta oscillations, we have such these co-occurrence. And the joint histogram shows various types of coupling distribution across pairs of frequency bands. After selecting the bands with strong coupling, we examine the coupling changes with the distance between electrodes, as you can see here. We found that in all cases, the coupling decreases with distance. Also, its value is stronger along anterior-posterior axes, as you can see in this case, but also the correlation coefficient between coupling and distance was stronger along medial lateral axis. Finally, by applying multidimensional scaling and T-distributed stochastic neighbor embedding on the coupling matrix of electrodes, and these two-dimensional coupling maps of electrodes were constructed. These maps are similar to the layout of the electrode array. It reveals that spatial patterns of spontaneous coupling can predict the anatomical arrangement. At the end, thank you for your patience and attention.
Great, thank you. Are there any questions? We have lots of time for discussion, which is great.
Yes.
So maybe I get started. Were the animals doing any particular task? I might have missed that.
No, at this experiment, our rats are anesthetized.
Oh, sorry.
And we just process the signals from spontaneous activity.
I see. Okay.
Yes.
So in terms of so you had a whole array of electrodes, which is really cool. And you see a particular spatial pattern in terms of the different coupling measures?
Yes, of course. In the coupling, when we assess the coupling with distance, we saw that in all cases the coupling decreases with distance, and actually we had some peaks on the patterns, but at the moment I don't know why we have these peaks on these patterns. And in the phase coupling, we have 3 clusters of electrodes specifically.
So can I just ask, when you talk about distance, what is distance exactly here?
Distance, I mean distance between pairs of electrodes.
Oh, I see.
We compare the coupling of pairs of electrodes with the distance between them.
Right. And is that dependent on where the electrodes are in cortex? Because you can have like same distance, but in different locations, for example, of recording, right?
Yes, from these coupling maps of electrodes, you can see, for example, the blue electrodes in functional maps are near together, and in the electrode array we have blue electrodes near together too, and these are the same from the functional map and the electrode array.
How far are those electrodes apart?
The distance, the unit of distance between electrodes, you mean?
Yeah.
It's about 400 microns.
Microns, okay, cool.
Yes.
Jacob, you had a question?
Yeah, are these within one barrel field, or are these across barrel fields? Do we have any information on that?
It's across barrel cortex, yes.
What is like the how many barrels do you think we might be on? Just sort of a scale.
The whole barrels are under the electrode array.
I see. Cool. We have a question from Muhammad. Have you compared wakefulness versus anesthesia effects? If not, then what effects do you think should exist in wakefulness that is different with anesthesia?
Excuse me, I can see the question from box.
In the Q&A?
Yes, in the Q&A. Oh no, I missed the...
So, should I read it again? So the question is whether you have compared wakefulness versus anesthesia. And if not, what do you expect would happen?
Yes, after this experiment, we may test this. In this experiment, we didn't test the wakefulness, and maybe after that, I complete this.
Do you have any predictions on what would happen or what would be different in awake behaving animals?
Yes, I think the coupling would be stronger along when we have wakefulness with simulated with the simulating of the whiskers. We have stronger coupling of in our results, and actually I think we won't have any decreasing of coupling vs distance. It's my idea at the moment.
Can you explain why you think that?
Because when we yes, because when we use stimulating all of the whiskers together, all the populations in the barrel cortex start to work and produce signals that all of them would be same together, and so we have synchrony in all across of the electrode, signals of electrode, and it would be happen, I think.
Cool. Sylvia asked a question, but I feel like we already answered that. Do you have any plan to test animals during behavior? I think you said yes. Is there a particular behavior that you would be looking into, or just having the animal awake and not do anything in particular?
And we use, in this case, a rat, but I think in other cases, we will see the same patterns. Our goal was that our goal was that assessing the mesothelial scale of population of neurons in human brain, but we can't use multi-electrode array in brain of human. So we use a rat to, after that, span our results for human or other mammalians.
Do you think there's any way to ensure that your findings translate to humans?
I think it can help for this research, but actually it is not completely okay for this case.
Yeah, I was thinking like Barocortex has a very particular organization, and I'm always a bit
yes, of course.
Yeah, a bit scared when people make claims about human physiology directly off that, because it's quite different in humans.
Yes, of course.
Awesome. Is there any other questions? Any of the other panelists, maybe? We have a couple more minutes. So anything else you want to tell us, Fateme?
No, thank you from you and from Normat.
Awesome. Thank you so much. That was a great presentation. We have 1 minute left, but I think we can just slowly transition here to Faize Amin Mansour. So Faize is, like myself actually, an engineer and physicist by training. So welcome to the club here. Lots of neuroscientists, in particular computational neuroscientists, have exactly your background. Like Conrad Kerding, for example, is the same. And of course, now she's working in neuroscience and actually looking for a PhD position. So if anyone is out there that particularly like her talk or her presentation, maybe this is a good time to get in touch with her. So in general, Faize is interested in questions about human and mental and cognitive capabilities, and also development of intelligence. And today she will be talking about the collective response of oscillating neural population to an applied perturbation, and that is based on a computational framework. All yours, Faize. You have to unmute yourself.
Okay, thank you for the kind introduction. So the question that we were interested in answering is basically related to understanding better the communication between cortical areas. So we know that all the fabulous cognitive behavior that our brain is capable of doing is based on some communication and some signal exchange. A common phenomenon between all of this is that how a network would answer to an incoming input. To approach this question, we take into account the oscillatory behavior of the cortex, which have been seen in different scale, like LFP, EEG. The spikes are coupled to certain phase of LFP oscillation. And we frame our question as follows: what will happen to the activity of the network if an incoming perturbation arrives at different phase of an oscillation? So for that, we used a well-known framework in physics, which is the phase response curve. a brief explanation about that is that if you take into account any physical oscillation with period of T0, if you apply a brief perturbation to the oscillation, to the oscillator, the subsequent cycle of the oscillator might get advance in its timing of happening, or get a delay, or remain unchanged. So this change in the shift is normalized by T0 and is defined a quantity called resetting. And when we plot the resetting with respect to the phase of the incoming perturbation, you will get a graph called phase response curve, which is a characteristic feature of the oscillator. So based on this, people define two types of neurons. Type 1, like intergalactic fire neurons, are always experiencing and advancing their activity. And type 2 neurons, like Hodgkin-Huxley, are experiencing both delay and advance, depend on the timing of the arriving input. So we used a network. So we use a network of LAF neurons, and you can see that LAF neurons are always positive in their phase response curve. So we used a network of LAF neurons, a recurrent network, which are connected randomly, and they were relative. The connections were sparse or relatively sparse. So 500 neurons were taken. The synapses were current-based, and they were modeled using delay difference of 2 exponentials. Then we applied an infinitesimal pulse to the network. And by infinitesimal, I mean the duration of the perturbation is 0.5 millisecond, and the amplitude was from the order of 1 millivolt. And we applied that to just 10% of neurons. And so we want to see what the collective response curve of the population of neurons would be. So before showing you the results, I would like to give you some intuition about the dynamic of the network. Here in the top graph, you will see the scatter plot of 500 neurons. In the middle graph, you are seeing spike density function. So the vertical axis is firing rate of the network. And the bottom graph is the LFB of the network, and the horizontal axis is time. So I will apply perturbation on different phase of oscillation between these two peaks, and let's see how the network is changing its activity. So first of all, I would like you to pay attention to the subsequent the time ahead of itself. Here you're seeing like 100 milliseconds ahead of that small perturbation, and you can see that different pattern of activity would arise. Sometimes there's more tendency to discharge in a synchronous pattern, sometimes less synchronous, and in fact they have tendency to discharge asynchronous. So and I'm showing you just like 100 milliseconds ahead. This duration is longer actually. Then I would like you to see this change in the amplitude of the subsequent oscillation, as well as its the phase shift, delay or advance. Then take into account that bump, which is moving with respect to the perturbation, and the whole manipulation of the LFP can be seen in the future of the network. So I think that's pretty cool. And let's see what's the results of collective phase response curve of the network. I take 30 trajectories coming from an ensemble of 5 networks, and this is the average value and standard deviation. So you can see that in the period of 2, in the complete period, the collective phase response curve is experiencing a delay at the early phase if the perturbation is applying at the early phase, and an advance if the perturbation is applying at the late phase of the oscillation. Also, in the amplitude response curve, you can see that initially the amplitude of the next oscillation is getting a decrease, and then followed by an increase, and again followed by a decrease for the second time. So why is this the mechanism? Why is this happening? So consider that a population of inhibitor networks work like this. A random set of neurons are discharging simultaneously. And then the discharge of these neurons induce an inhibitory signal to the old network. This takes time until the overall activity of the network get back to its resting state, and then in the second window of activity, another random set of networks are getting discharged. When we apply perturbation here, for example, the number of neurons which are getting discharged are getting enhanced, and this means that the overall inhibition of the network is increasing, leading to the network requiring longer time to getting back to its next oscillation, which is equivalent to creation of a delay in the occurrence of the next oscillation. At the same time, because the discharge is taking place in a shorter period of time, the network has a tendency to be to discharge more synchronously, and that is why the activity of this next oscillation is getting enhanced. So then, if we take into account a perturbation in this spot, here still the perturbation leads to an increase in the inhibition of the network, which leads to a negative value for PRC, and but because it is applied in the middle of the period, the overall network is experiencing smaller synchrony, and that is why the amplitude of the next oscillation is enhancing. And finally, in the C, if the perturbation is applied in the C phase, here this is the time window where the network is in its maximum excitability. So if the perturbation is arriving here, all the neurons which were supposed to discharge here are getting advanced in their discharging, and so the PRC has a positive value, and amplitude also getting enhanced because of that perturbation and the small time window that the perturbation is applied. So what I showed you is that timing of variable input matters. It affects on the future pattern of the network discharging regime. It affects also transiently on the frequency of the network because of the delay or an advance in it, in the subsequent cycle of oscillation. And in the synchrony of the discharge of the subsequent cycle of the network activity. Also, I showed you the collective PRC of the population is independent of the PRC with single neuron. We talked in detail about the pattern of the collective PRC and amplitude and collective amplitude response curve. And a falsifiable prediction of this model is that if I apply a perturbation at early phases of SFT oscillation, which has an equivalent for LFP oscillation, this gives rise to transiently reduction of network frequency, while applying in the late period of oscillation induce a transient increment in it. So I would like to thank my collaborator, Dr. Ali Reza Alizadeh, who was my first mentor in neural stimulation. And thank you everyone for attending. And I'm happy to take questions.
Great, thank you so much, Faize. We have a question from Mohammed. Did you measure any complexity index, such as Temple-Ziff or entropy measures of LFP modeling in response to perturbations in different phases?
No, actually, we didn't do that, but I think it's a good idea. I think we should do that too.
I have some, while other questions are coming in, I have some very basic questions. So first of all, how did you ensure that you get your network to oscillate?
So this network that I described for you here is a well-known network mainly introduced by Brunel in 2000 and a couple of more papers are on it in 2003. These networks are, the dynamic of these networks is what I explained to you. And that is a random number of neurons are getting discharged, and they induce that inhibition to all the network. The overall current of the network reduced, so no one can discharge. There's a silent period, then there is an enhance again, another random subset of network getting active. So this is
Cool.
the way that this network is working.
We have another question from Joao. Great talk. Do you have any prediction on how different perturbations in excitatory inputs at different phases would affect different dynamical systems like Wilson-Cowan or coupled Wilson-Cowan models?
I was thinking actually on considering those models too, but so far, no, I have no idea how it might work on those models.
So one of the things I found really interesting is the fact that obviously the brain is known to oscillate, but we have very little knowledge about how these the relatively low frequency compared to the spiking oscillations affect the spiking behavior of neurons, right? Can you maybe say something about how this relates to spiking?
So I'm not sure if I get your question completely. These oscillations that I showed you are in the range of gamma.
So typically, like a lot of oscillations in the brain are much more low frequency, right? So we would have like 10 hertz, 20 hertz, so alpha and beta band oscillations that are believed to carry like sensory and motor signals, or at least correlate with them. Can you maybe say something about how these oscillations, based on your results, might influence the way that the brain processes?
I had another simulation, which was a study on those low oscillations when there are still that high oscillations exist in the network. And actually, I was working on the coupling, phase response coupling of the low and high oscillation. So what I learned from that study was that the slow oscillations are results of synaptic activity. In other words, there are if you measure the synapses in the model, if you measure the for example current of each synapses, you will get a very big power of low oscillation on them. But most of them are disappeared due to collective activity of neurons, because they have different phase of activities. But at the end of the day, a small low oscillation would remain, and actually those small oscillations had phase amplitude coupling with the phase amplitude of the high frequency. So what I know is that, yeah, these are the effects of synaptic activity.
Great, thank you so much. We have to stop here. Mohammed asked whether the code is available for further work and collaboration, but I'd encourage you to contact Faiza directly through Twitter maybe or by email, and then and everyone else who has still open questions can do the same thing. So with that, I would like to thank everyone again for the great session. Thank you, Jacob. Thank you, Fatime. Thank you, Faiza. I learned a lot, and I enjoyed the session very much, and I hope so did you. See you soon.
Okay. I think we can get going, Ariana. What do you think?
Yes, perfect. All right, then.
I can share a screen or?
Yeah, so first of all, I just want to everyone that is joining us and following us on YouTube, say hi. I'm Valentina, and together with Ariana, that will be our amazing helper behind the scene, we bring you a great lineup of speakers for this session. And each of our speakers will have 12 minutes to present the research, followed by 3 minutes for your questions. So please write your questions in the Q&A section that is at the bottom of the screen, and keep them coming during their talks, and will be my pleasure to read them at the end. But also from time to time, check the chat, because Ariana and I will be sending link and interesting messages there. Starting with a couple now about our speakers. Great. So let me introduce the first speaker, Maria Coneva, which is a last year PhD student in the Machine Learning Department and Neuroscience Institute at Carnegie Mellon. She's co-advised by Leila Webe and Tom Mitchell, and she will be presenting us her work on modeling task effects on meaning representation in the brain via zero-shot mag prediction. So Maria, feel free to share your screen, and I'm gonna mute myself.
Great, can everybody see my screen? Awesome. Yeah, thanks a lot for the introduction. Thank you. And thank you for having me to NeuroMatch, and for you guys joining today. So yeah, today I'll tell you about the recent work on modeling the effect of a task on the representation of meaning in the brain. So previous studies on meaning representation have shown that the brain accesses meaning differently depending on the demands of the task. So for example, if I show you the word bear, under two conditions, in one condition I ask you whether bear is a vegetable, and in the other I ask you whether bear is a tool, your brain activity while seeing bear will will show this thematic difference due to this different task. And this has been shown by previous work in neuroscience. So one hypothesis for how this task influences the brain activity is that in order for the task to show this influence, it uses attentional mechanisms that emphasize some aspects of the stimulus that are task relevant. So for example, some mechanisms that can do this are increased response gain, sharper tuning, and a tuning shift. But really, the computational mechanism behind this attentional effect is not really well understood. There are all these hypothesized mechanisms, but there's not really one answer. So what we set out to do was to provide a framework which can be used to test different hypotheses for the interaction of the task and the stimulus in the way that they give rise to the brain activity. So we give this computational model that predicts the brain activity given as a representation of the stimulus and the task. And the work was recently accepted in Europe, and we're working on the final version of the manuscript right now, and will be available soon. But in the meantime, you can check out my website for a version of this paper. And we've also provided the code, so if you're interested, please take a look there. So to begin, let's take a look at the data set that we use. So here we have MEG recordings of 6 subjects for answering questions about different nouns. So we have 60 nouns in total and 20 different questions. And here we think of each question as a different task. So the data was originally collected by Gus Udre. And in the original paper, they tried to answer the question of how are different noun properties processed in the brain. So they used decoding analyses on the original data, which was averaged across different repetitions of the same stimulus under different different questions. So this unfortunately lost the task-dependent information. So here we're going to reanalyze this original data on the single trial basis because we're interested in this interaction between the task and the noun. So in our analyses, we're going to predict image recordings as a function of both the stimulus and the task. So in order to do this, we need a representation of the stimulus and the task. So one way to get these representations is by thinking of them as just words. So we can think of bear as the word bear, and the task of answering the question, is it man-made, as the words, is it man-made? And we can provide these words to a natural language processing model, which then gives us a representation of these input words in the form of a vector, a numerical vector, which we can then use further in our analyses. So indeed, this is one way that we obtain these representations. And we use BERT, which is as the NLP model, which is state-of-the-art natural language processing model. Another way to get these representations is through mechanical Turk, where people respond to similar questions about 120 nouns. And I'll tell you a little bit about this representation in a second, but first I want to tell you about how the mechanical Turk representations compare to the BERT representations. So in terms of the stimulus, it doesn't really make a difference whether we represent it via mechanical Turk or BERT. Both perform equally well at predicting the imagery recordings. However, for the task, it does make a difference. So the mechanical Turk representations actually do much better at predicting the brain activity than the BERT representations. And we think that one explanation for this is that BERT may lack some common sense reasoning knowledge about object properties that are necessary to answer the questions that we have. And in fact, some prior work has shown this about BERT. And we've included more experiments in the paper. So if you're interested in this difference, please take a look there. But for the purposes of this talk, I'll be focusing on the mechanical Turk representations. So how do we obtain the stimulus and task representations given this mechanical Turk answers? So again, we have 218 questions and 120 words. And 60 of these words and 20 of these questions actually occur in the experiment, in the MEG experiment. So we're not going to use these mechanical Turk responses to obtain the representations. We're going to use everything that's not in the experiment. So in order to construct a representation for the word or the stimulus, for example, carrot, we're going to use the mechanical Turk responses for all questions, all 198 questions that do not occur in the experiment. And similarly for the task, in order to represent it, we're going to use all 60 words which are not in the experiment and the mechanical Turk responses for these words to that specific question. So this representation for the stimulus was actually previously proposed by Gus Udre and his colleagues and was shown to be predictive of the image recordings. But the task representation is something that we formulated. So we wanted to make sure that semantically similar questions are also close in their task representations. So in order to check this, sanity check this, we can look at the pairwise cosine distance between the task representations. And here, dark colors means low cosine distance, which means two tasks are very similar. And we can see this block diagonal pattern, which corresponds to roughly semantic divisions. So the first block corresponds to questions about manipulability. The second one is about object properties, such as size. And the third one is about intimacy. So here we can see that the task representations do contain some kind of semantic information about the question. So now we have representations for both the task and the stimulus. So we want to formulate hypotheses about how the two are joined together to give rise of the brain activity. So first we have two very simple hypotheses that we can use as controls or baselines that we don't expect to do well or as the best at predicting the brain activity. So the first hypothesis is that the stimulus is the only thing that affects the brain activity. And the second one is the task is only affecting the brain activity. The third hypothesis is that both the stimulus and the task affect the brain activity. However, their contributions are independent of each other. And lastly, we form a hypothesis that is based on the attentional task effect that we discussed in the beginning. And so here a task emphasizes specific properties of the stimulus that are relevant to the task. So for example, if the task is it bigger than a microwave, it may emphasize properties of the stimulus that are related to size. And so here, this function G in this hypothesis 4 can be learned. Or we can also, if we have interpretable features for the task and stimulus, we can also pre-compute this function G with some kind of hypothesis that we have. And I'll show you exactly this in a second. But one more note about the training of these hypotheses. So here we model F as all of these functions F in the 4 hypotheses as a linear model regularized by the Ridgepan OT. And there's more training details in the manuscript. But basically, in brief, we train this in a cross-validated way, and we test on held out samples. So next I want to show you just briefly what this hypothesis 4 can look like when we want to encode a pre-computed relevance of the task for the stimulus. So first we need to think, how can we determine how relevant the task is for each stimulus dimension? Well, for us, each stimulus dimension is actually a task, if you recall. So the relevance for a task for any particular stimulus dimension can be computed as cosine similarity between 2 task embeddings. So to give you an example of what that relevance looks like for a specific question, so if the task is it made of metal, the 5 most relevant dimensions are: is it silver? Is it mechanic? Was it invented? Is it shiny? And does it have a hard outer shell? So we see here that the stimulus dimensions are indeed semantically close to this task. So we can use these relevance scores, the resulting relevance scores, to weigh each stimulus dimension according to its relevance for a specific task. And we can use this as the augmented stimulus now that we can then predict the imagery recordings with. So let's see how these different hypotheses do at predicting the imagery recordings. So here I'm going to show you a performance metric, which is called 2v2 accuracy. And this has been previously proposed for settings with low signal to noise ratio. So you can ask me about this later, but I think for these purposes, it's enough to know that the theoretical chance performance is 0.5. So now I'm going to show you the average performance of each hypothesis across all subjects and sensors per time point. So for the 2 simplest hypotheses, we have the blue one is the stimulus-only hypothesis, and we see that it starts to significantly predict the brain recordings around 175 milliseconds, up until 650 milliseconds. And the yellow one is task-only hypothesis, which mostly predicts the image recordings around 500 milliseconds. And now we want to add all the other hypotheses, which all include the stimulus and the task. And so these are shown in green, red, and purple. And we can see that they all perform similarly, but they also perform better than the hypotheses that include only the task or only the stimulus. So in fact, the pre-computed attentional hypothesis that's shown in purple significantly outperforms the stimulus-only hypothesis around 500 milliseconds. So we can see these results, but they are averaged over all sensors. What sensors actually show this task effect? And we can see this by plotting the performance across individual sensor time points. And we visualize the question and word effect separately here. And we see that the main question contribution on the left is actually in the frontal and temporal regions during 400 to 550 milliseconds. But on the right side, the word effect is more distributed in time. And we see that it's mainly occurring in the occipital and temporal lobes. So one takeaway from this is that the question effect seems to coincide with the end of semantic word processing. So in conclusion, we showed a computational model that predicts both predicts brain recordings, both as a function of the task and stimulus semantics. And importantly, we trained this and evaluated in a zero-shot setting, which means that we can generalize to both stimuli and tasks that don't occur in the experimental setting, as long as we can represent them, of course. We also showed that the models that actually integrate both tasks and stimuli predict the imagery recording significantly better than those that only incorporate one of these, and that this task effect occurs at the end of semantic processing. So we hope that our work can provide a framework to formulate hypotheses for how the task affects meaning representation. And our results, I think, pose a challenge for future work to formulate new hypotheses that can show earlier effects on the task and the semantic processing of a word. So thank you so much for your time, and thanks for NeuroMatch and the opportunity to speak to you today.
Thank you very much. Great talk. Extremely interesting. And I'm going to give people time to write up their questions, but I remind you, we only have 3 minutes. So everyone else should just look up for you online and check out the paper for the details. But maybe just to get started, I do have one question, which is kind of classical. It's about your next step. Because as you mentioned, this is a framework that can potentially be applied to a lot of different settings in neuroscience. So do you have an ideal data set in mind already?
Yeah, that's a great question. I think one limitation that we saw is just power. So we had few subjects and kind of little data to train our more complicated models. Like I mentioned, this fourth hypothesis had a model where you could learn how the task augments the stimulus representation. And we showed that we have too little data to really learn this well. So it'll be really great to have more data sets that have this different tasks in them. I think, unfortunately, in neuroscience, I've seen data sets that have a few task conditions, but not as many. So I am interested actually in collecting more data sets that have more of these varied tasks. Like, for example, in this paper we had, in this data set, we had 20 different tasks. So that would be great to have more of, perhaps more subjects. And also, I think actually more nouns per task would also be very helpful.
Right. And would you dare trying with tasks that are more different one another? Because in this case, I understand the principle of considering each question a different task, but it's still sort of the same, right? Would you?
Yeah, for sure. I think that's a very interesting question. Yeah, I think as long as we have a way of representing not only the task semantics, but also kind of what the task requires, right? I think as long as we have that, we can use this kind of framework. But that is sometimes challenging if we cannot represent what the task is asking us to do. So yeah, the question tasks that we have are quite simple. But I think, yeah, show that this first step towards incorporating the task effect for sure. But yeah, definitely a next step would be to include more varied tasks and things that maybe are beyond yes or no questions also.
Yeah, a lot of data collection that will hopefully resume as soon as we can start scanning again.
Yeah.
Oh, thank you very much. Yeah, and we will be sending you, if someone writes questions later on that are pertaining your talk, we will be sending to you.
Awesome. Yeah. Thanks a lot.
Thank you. And with this, we can move to our next speaker. Let me make sure that I don't get it wrong. Perfect. Our next speaker, Sebastián Bobadilla Suarez, who is a postdoc in the Department of Experimental Psychology at UCL. working with Prof. Brad Love, and he will be presenting us his work on the neural similarity in bold responses and multi-unit recordings.
Cool, thank you. Can you see that? Okay, I'll start my stopwatch. Thank you everyone for coming to see my talk on neural similarity in fMRI and multi-unit recordings. Thanks to the organizers of NeuroMatch and Ariana and Valentina for hosting us. So I'll present work in collaboration. Well, my name is Sebastian Bobadilla Suarez. I'll present work with Brad Love. I'm in his lab at UCL, and a master student, Marilena Lemonari, and supported by Wellcome Trust, Royal Society, and NIH. It's also in collaboration with Earl Miller's group at MIT. Well, Marcus Siegel's at Tübingen now, but he was at MIT when the work was done. So yeah, to get started, well, how does the brain represent similarity? That's what I'm interested in. So just to give an example, there's a person in a scanner, you show them the picture of a robin, right? So you get a brain activation, and that's the representation that we can work with. You can do the same for a sparrow, right? So you can compare the brain activations as opposed to the images themselves to see how the representation differs for each one of these birds. And then compare that to similarity with the robin and the parakeet, for example. So then we have sets of similarities, right? And then we're interested in quantifying these similarities at the brain activation level. So how do we pick these similarity measures? Because each measure has its own assumptions, right? It'll focus on different aspects of the data, of the brain data. The way we pick the measure, the one that best describes the brain data, well, it'll reveal something very fundamental about the neural code in a way, you know? Each measure, like I said, has hidden assumptions that has mechanistic and theoretical implications of how the brain is representing stimuli. So the first part of this talk is fMRI in humans. And in that study, it's published already in Computational Brain and Behavior. We focus on a set of measures that we're interested in. Each family of measures focus on different summary statistics of the fMRI data. So some will focus more on like an angle type information, or maybe the magnitude of the numerical vector. So we're thinking numerical vectors here for voxels, or maybe more distributional information. So just to give a quick example, like this is a similarity measure like Euclidean distance would say that objects A and C would be more similar here, whereas a measure that focuses on angle would say that A and B are more similar in this two-dimensional voxel space. A common measure that people use in this type of studies is Pearson, for example. But I think we have to question the choice of measure to really tell us something about how the brain represents stuff. So like I said, this is published work. The work actually focuses on two previously published fMRI data sets, one from Mike Mack and Ali Preston, the other one from Optabeek and Stephanie Bracci. So it's two different stimulus sets that we're interested in. One that just, in their task, they just show geometric shapes in this data set here. And in the other one, just natural images. So it's different stimulus sets. And we're interested to evaluate the measure in each of these data sets to see if there's differences between the data sets, but also between brain regions. Since we're interested in these differences between brain regions, you know, we can't really just rely on behavior, because different brain regions might have different measures that they like to work with, or depending on the task as well. So it goes well with what Maria was talking about in her talk about this interaction between the stimulus and the task demands. So for this methodology, the logic is that similar stimuli are more easily confused, very intuitive way of thinking about things. So we can use a classifier to decode these brain activations to differentiate between a robin and a sparrow, for example. And we can use the confusion matrix of the classifier as our reference, as the gold standard of similarities. We can compare that to the other similarity matrices that we're interested in, like Pearson or Mahalanobis. And we constrain this to areas that have good decoding accuracy, so that actually have enough information for us to make this comparison. And just to keep it short, I'll just say that in the top row, we have these geometric shape data. So this is more of a visualization, but in blue is the Euclidean measure, and in red is the Mahalanobis measure. And just in general, yeah, we see that Euclidean type measures and magnitude type measures work well with the geometric shapes data set. Whereas on the bottom row, Mahalanobis totally dominates in the natural images data set. So if you want more information, you can look at the paper on that. Just to pivot a bit here, this is really exciting work that we're doing with Errol Miller's data on multi-unit recordings of macaques. And what does this bring us compared to what I just showed you? Well, here we can look at more temporal resolution, right? So look at the temporal statistics of the neural activation and see what we can find there with the same kind of logic that I was talking about. So it's a hotly debated subject, whether it's rate measures or temporal based measures for spike trains that are important. But yeah, like I said, at least we can do this comparison here, and you can't really do it with slow fMRI data. So it's based on this paper in Science from Errol Miller's group, if people want to take a look. So in that paper, well, just to mention, the data has the advantage that, just like Maria was talking about, the task demands will emphasize different dimensions of the stimulus space. And that's something we're really interested in as well, just because that basically also tells you something about the representation and the similarity between the stimulus, what dimensions are being emphasized in the representation. So the monkeys, Rex and Paula, and that's not them, but anyways, they did thousands and thousands of trials discriminating random dot motion. But before they see the random dots, they would see a cue to tell them if they have to discriminate the motion or the color. It was 100% coherence for the dots. And we can see in panel C here, that's the actual representation of the stimulus space. So you have color on the y-axis and motion on the x-axis. And this is obviously, it was defined by the experimenters, so hand-coded in a way. And then, yeah, you give a different response, left or right, depending on if it's motion or color that you're discriminating. Well, Rex and Paula in this case. And it's a massive data set. It's really nice data set. They looked at did like 6 areas in the brain, sometimes simultaneously, over many sessions. So really cool stuff. Like I said before, here we can actually look at the spike trains, right? We have different neurons with different spikes, different time points, and we have these different areas to work with. So just at a broad scale, we can see, okay, is timing information relevant, or is the average firing rate what's relevant, as people normally talk about? So what summary statistics should we consider? We chose 3 measures for each class of measure. On the left-hand side, we have ISI, which is about the interspike interval, spike and sync, which I won't really go into too much detail, but I'll give you the reference. And then the ones we talked about before, Euclidean, Pearson, Mahalanobis, defined on a certain area, brain region or lobe. So that's the reference for the timing measures that we took into consideration. So yeah, just to remind you, this is the stimulus space that the monkeys were working with. So each stimulus here in this graph was basically represents the random dots that they saw at some point. And they would have to give a left or right response depending on what they needed to discriminate, color or motion. Just to make, again, the long story short, I just want to clarify here that the logic is a bit different in that before we were using a confusion matrix of a classifier to infer like the gold standard similarities to compare with. In this case, we're using the ones defined by the experimenter. So we have these stimulus coordinates that I just showed you back here, right? These are defined by the experimenter, and we can use that to get our similarities as our gold standard, if you will, and then use that to compare all the other measures that we see in the empirical data. And what we see is actually that this ISI measure does relatively well, quite well compared to the other ones. In general, the timing ones seem to do better than the firing rate ones, but ISI just really has good performance. Something else that we were interested in, it goes well with what Maria was saying, is that the task demand, so whether you're discriminating color or motion, it'll change the representation. We've seen this in human data, for example, or in monkey data. And just from the behavior, for example, you see that the representation stretches along the relevant dimension. So if you're discriminating color, it'll stretch the y-axis. Complementarily, if the monkeys have to discriminate motion, you'd expect the stretching along the motion axis. This helps the monkey solve the task because it makes the discrimination easier if they represent things this way. And this has been shown also in fMRI. I'm not sure if you can see that reference, maybe it's cut off. But anyways, by Mac, Love, and Preston and PNAS, if people want to check it out. So with humans, they had a category learning model that would look at these emphasis on the relevant dimension depending on the type of problem. So you would get these attentional markers of the stimulus representation, And that would be tracked in hippocampus, for example, in that paper if people want to take a look. But again, the stimulus was hand-coded by the experimenter. So you're kind of already guessing what the representation is like, which is why we tried to actually guess it with a classifier in the first part of the talk. So yeah, that's just what I said right now. So how to get at, oh, sorry. Anyways, I don't know if it's cut off at the bottom, but basically to get around this issue of hand-coded representations, another strategy is to try to infer them with a model that learns to do the task as well. So in this case, we can use a deep learning model. So this model, it was done by Marilena Lemonari, a master's student. It doesn't have access at all to the neural data. So all it has access to is the same picture that the monkey saw to actually do the task. So the pixels and the label of the stimulus, depending on if they had to discriminate the motion or the color. So it's basically has to learn what the monkeys were doing. We basically got a CNN that was pre-trained, VGG16, fine-tuned it to the images that the monkeys saw to discriminate the images. And then we got the penultimate layer of activations, and we fed that into an LSTM. So it's another type of learning model that's good for sequence learning. And basically, yeah, the model learned to do the task. You know, it basically learns to do cognitive control, provided only with that supervision signal, the stimulus labels, and a pre-trained visual system, which, you know, the monkeys also have a pre-trained visual system. So yeah, I thought that was pretty cool. The model also stretches the dimensions along the relevant, along the relevant dimension, depending on what they have to discriminate. So just take home messages. So similarity measures can differ across tasks, but in the fMRI, we see that they stay constant across regions. And they can benefit from timing information. That's what we see in the multi-unit recordings. And they can also, you know, across these different modalities, the model, the LSTM model, fMRI, multi-unit, they can provide a window on the stimulus dimensions, on the stretching, you know, depending on the task demand. And in this case, you know, it's a cognitive control process based on task demands. You get these attentional effects. And yeah, I'll just give my contact information, and thank you to my collaborators, to everyone who organized and our funding. And yeah, if anyone wants to contact about me about anything, I'm more than happy to chat.
Thank you. Thank you very much for presenting this extremely interesting method that, as you said, has a lot of connection with what we heard before, and it definitely can be applied. And I already see a question that Benjamin Silver is asking, so I'm going to read it for you. Has anyone ever compared similarity data from a classifier to self-reported similarity data? So for example, a subject that rates a parakeet on a scale from a robin to a sparrow, and investigating what might cause varying levels of divergence between the two,
So yeah, I don't know off the top of my head, but yeah, definitely people have looked at behavior. You know, that was what was normal before. As far as using a classifier on a region, I actually think that the fMRI data set we were basing on from Optabeak Lab and Stefania Bracci, they actually look at something similar to this. I don't know if that answers the question, but yeah, definitely. Yeah. I don't think anyone has actually tried to focus on just inferring it directly from the neural activity itself. Because behavior will only tell you something about that final stage, that output stage. The different brain regions might act differently, although that's not what we see, right? in their data.
Right. We have another question. Yeah, and we have time. Jules Burchard is asking, would dimension stretching relate to the dynamic range adaptation and efficient coding?
Oh man, dynamic range of adaptation, efficient coding. I'm not sure. Sorry, I'm not sure I understand that question. So are they saying that there might be like a cap on the stretching? I'm not sure.
Or at least a relation between how much a given dimension would be stretching for a given subject and how much that subject is.
I'm not sure how to answer that question, because stretching is obviously a relative term to the other dimension. I don't know how you can measure it like in an absolute sense, the stretching, because it's always the ratio between the scale of dimension Y versus dimension X. So it's the problem you always have with attention, is that one dimension is being inhibited, or is that the other dimension is being tuned and you got to gain response gain on this other dimension.
So I'm not sure how you would answer that, but I have to think about it. Yeah, thanks.
Right. Yeah, great question, because it's triggering a lot of reflections in our audience. So thank you very much for your talk.
Thank you.
Yeah, please feel free to reach out to him online. And
cheers.
We can move to our last speaker. We have Stephen Patterson, a postdoc at the University of Washington in the lab of Brenton and Rao, and he will be presenting us his work on transferring generalized neural decoders across participants and recording modalities. So please, Stephen, take it away. We cannot hear you, sorry.
No, sorry about that. Thank you. No. All right, so yeah, I will be talking about creating generalized, robust neural decoders. And so the motivation for this is brain-computer interfaces like this, and also similar ones like this, where you sort of take neural data that you record and interpret it in a way to control some sort of device. And there's a lot of really cool things you can do with that, but there are some limitations in terms of you need a lot of training data that can take a long time to get. And you also may need to recalibrate frequently because the data you record is non-stationary. So the problem is that we have these great decoders that we want to train, but the amount of training data we have is really limited and can be very time-consuming to obtain. So a solution to this is, instead of trying to get a lot of data from one participant, is to get a little bit of data from a lot of participants, basically. And then you can create this decoder that takes in all of that data and finds some representation that works across all those participants. But it does have to be robust to differences across participants. And I'll get to that in a second. So the input data I'll be talking about here are neural population recordings. So thousands of neurons, tens of thousands of neurons. So I'll be talking about ECOG, which is electrodes on the surface of the cortex underneath the skull, and scalp electroencephalography, or EEG. And so ECOG has a really nice high signal to noise ratio. However, it's invasive, and so it requires surgery. So we only look at this usually with clinical patients. Whereas ECOG is really commonly used in research. However, it's very much prone to artifacts because it's above the skull and can move a lot. So back to what I was talking about with the generalized decoding and how we can create these participant differences. One of the things that can differ from one person to the next is the frequency content of the signal of interest. So here I'm showing a spectrogram, which is basically in the frequency domain of your signal, average across 12 participants. So it's very similar if you're doing like a Fourier transform. In this case, it's wavelets. So the x-axis here is in time, where 0 is when the participants move their contralateral arm, and frequency is on the y-axis, and then the color scaling is in decibels and indicates the magnitude of the spectral power of the signal. Basically, what I want you to take away here is that there's these two very distinct bands of activity, this low frequency power decrease and this high frequency power increase. But if you look across participants, you actually see that there's these differences. This pattern is not conserved across all participants. So what you really like to do is figure out what features are robust and consistent across all participants when you're creating a multi-participant decoder. Additionally, particularly for ECOG, because it's clinically motivated, the electrodes are placed based on clinical need, and there's no standardized placement. So this is showing 4 ECOG participants colored differently. And you can see that there's not necessarily a great way to always align electrodes among participants. That's another challenge that we want to address. So the main idea is take data from multiple participants, train a generalized decoder, and then also we can take just a little bit of data from some participant we've never seen and fine-tune that generalized model using much less data than we had before. All right, so first I'll talk about the generalized decoder. And so similar to what Sebastian talked about, I'm using a deep neural network, in this case, convolutional neural network, because it generates data-driven features. In this case, I'm looking at stuff applied directly to the neural data in question. So for this, we used EEGNet. This is a model created by Lawhorn et al. In Journal of Neural Engineering. It's a really nice paper if you're interested. Basically, it takes the EEG data segmented on the left, runs it through this decoder, and then outputs some decoded behavioral state, decodes your behavioral states. One of the reasons I really like this is the temporal and depthwise convolutions at the beginning are very analogous to a data-driven bandpass filter and a data-driven spatial filter, respectively. So to me, that's very understandable, and it's a very common pre-processing method when you do neural decoding. So one of the downsides of this method is so it can sort of sort out differences in frequency content, but it does it all in the time domain. And one of common ways to analyze neural data, at least from ECOG and EEG, is to look at it in the spectral domain, similar to that spectrogram I showed before. So to do this, I inserted a Hilbert transform here after the temporal convolution. And Hilbert transform is very similar to wavelet transform. It's like a Fourier transform, but it can handle data as that changes. So like here, you can see that the power of the signal changes over time, and the Hilbert transform on the bottom here actually picks that up and changes with it, which is good for non-stationary signals. This is also, with the temporal convolution, very similar to data-driven filter Hilbert method, which is nice. So that sort of allows for that. But the downside still is that this is made for EEG data, so the electrodes are assumed to be consistent, which is not the case for something like ECOG. So to get around that, we pre-computed these weight matrices between the electrodes, which vary across participants, and common predefined brain regions that are the same across all participants. And so you have to just pre-compute these, but you can insert them into the model and do a matrix multiplication partway through the decoder itself. And what that means is everything before the matrix multiplication step differs across participants, but everything after the multiplication step is on these same, same points across all participants. And so it really gives the decoder a chance to do a good job in decoding. So that's how we created the decoder. And now I'll talk about how we actually tested this and how it performs in practice. And so for this, we tested on real data. We looked at 12 participants who had ECOG, and this is part of our long-term clinical naturalistic monitoring data set that we have. That's part of the preprint in the chat. Additionally, we also looked at EEG from 15 participants, and this is from a paper by Offner et al. And +1. And for both of these cases, we were looking at decoding between rest, where they're not moving their arm, and move, where they're just sort of doing some sort of roughly elbow flexion. That's what they're doing in EEG. In ECOG, most of the movements they do look very similar to that. So it's just two-state classification. And in terms of experimental design, what we did was we took 11 of those 12 ECOG participants and trained/validated on those. And then we tested on withheld data from a withheld, from that 12th ECOG participant, and that's what we call the same modality condition. And then we also tested on the unseen EEG participants, and that's what we call the unseen modality condition. And of course, we also randomized, did a bunch of models where we randomized, which participants were training, validation, and test set, and did that 36 times and looked at the results to ensure or reduce any bias in how we did things. So first, for the same modality conditions, so this is going from ECOG to testing ECOG participants. So these are the accuracies we have on the withheld participants. So test accuracy is on the y-axis here. And then random chance is at 0.5. And so what you can see is HTNet does much better, significantly better than these other three decoders. So EEGNet, random forest, and also minimum distance. It's really important to note here that because this was trained across multiple ECOG participants, I actually had to include that projection step onto common brain regions, because otherwise this would just not work at all. There would be no way to align these things very well. So the only difference between HTNet and EEGNet is that Hilbert transform. And you can also see across ECOG participants, when they were the test participant, in general, HTNet seems to do much better than the other three methods. And then looking at unseen modality decoding, this is originally, when I tried this, I did not expect it to work very well at all. And for 3 out of 4 decoders, it kind of works as you'd expect. The scaling is different in some ways. The noise that you get is very different. And so the decoder can't really generalize without having seen anything. But in HTNet's case, it actually can. And this is mostly due to the fact that it looks in the spectral domain, and you can do relative power metrics that actually are very robust across at least EEG and ECOG modalities. And this is also very consistent across different EEG participants. Okay, so talked about how we can train those decoders, and now I'll talk about fine-tuning them. And so this involves taking a pre-trained model, so any of the models I just showed you, and then taking a little bit of data from that withheld participant that we're testing on, and then training on it a little bit to fine-tune it a little bit more towards that participant, and then testing again. And so this is work that another grad student in the lab, Zoe, has helped out a lot with and worked really hard to do. And so when we fine tune in the same modality condition, we're taking the pre-trained model from before and then training a little bit on that withheld participant and testing on the same data as before. And what you can see here is that even with just a few training events, so like up here, you get this really quick boost in performance that's consistent across participants. And so when this pink line here shows the performance of randomly initialized decoder, and these other ones are the fine-tuned ones. You can see that initially, as you might expect, randomly initialized decoders don't do so well, and over time they do pretty well. But even that performance you gain here with over 1,000 events is very similar to what you gain with a fine-tuned model with very few events. And you can also see something very similar for the unseen modality decoder. So yeah, so just to wrap up, showed you how we built this generalized decoder for neural decoding in both ECOG and EEG. Showed you how we can test that and how it actually outperforms other comparable decoders. And that you can also fine-tune this model, these pre-trained models, and not have to use too many fine-tuning events to get really good performance for each participant. And so there is a preprint that should hopefully be coming out within a week or so. Yeah, and I'd also like to acknowledge all the help from the Brunton Lab and Raj Rao's lab, who are both my co-advisors, along with all the clinical staff at Harborview who helped collect the data and work with the data, along with our funding sources. And I also want to thank you all for paying attention and listening. And if you have any questions, I'd be happy to take them. Thank you.
Thank you very much. Really, really interesting. I'm quite impressed. Honestly, I was not. Coming from fMRI and being so focused on topography most of the time, the idea of trying to go across participants that have a different coverage, just as an idea, scared me. So maybe could you elaborate a bit? It probably was already in your graph with the individual participant performance, but how much does the decoder performance vary as a measure of how similar is the coverage across participants in the end?
Yes, that's a great question. So I didn't show it, but basically we also did test that to see how much the training participants overlap with the test participant. Mm-hmm. And you can actually see that the decoder does a lot worse when there's not as much overlap, as you might expect. If you're not training on the area that you're interested in, doesn't necessarily generalize as well. But we found our decoder, when things overlap really well, just performs much better than these other decoders that we were looking at.
Right. And okay, so we see another question, and I should let someone else ask as well. So Joelie is asking, can you elaborate on why you choose to use a pre-computed projection matrix to account for different recording locations versus learning that end-to-end?
Yes. So in some ways, we had actually tried to do sort of a learning end-to-end. We had tried to do some matrix completion with this before, and it just did not work very well at all. That's basically the short answer. I think there might be a way to make it work. The nice thing about this is it uses information that we already have available and that we can get pretty early on. and sort of just constrains things so we don't have to learn as many parameters. It should take less time to train and be most likely a little more stable.
I see. Okay, we have another question from Karen Jerby. Is asking, was the random forest classification used with spectral amplitudes from the Hilbert transformed as features? This is probably needed for a fair comparison with your model.
Yeah, so that's a great question. The stuff I was showing here was using the time domain. I've looked at it with the Hilbert transform as well, and it, the performance, as you might expect, does improve. One of the things I was trying to stay away from with not doing the Hilbert transform in advance is you sort of need to know what frequency bands are of interest before you do it. And I think that's one of the advantages of the HT-NUT method is you don't really necessarily need to know anything about your data, at least about the specific bands in and of itself, because it can kind of learn those. But that is a very fair point. If you do know what you're doing, what you're looking for in advance, you don't necessarily have to do this.
Sounds great. I think we're out of time, but thank you very much to all three our speakers. It was a great session and a lot of points in common between the three talks. So really, thank you very much. Thank you for everyone that has been attending and asking questions and following on YouTube. And just enjoy the rest of the conference.
Thank you.
Thanks, everyone.
Thank you.
Bye. Bye.
Trying to bring people on screen to ask questions. So I could hold up a card like this to let you know when you have 1 minute left, or I could just interrupt you by talking. So whichever you prefer, just let me know, and I can interrupt you so that we have a lot of time for discussion.
I prefer the card, and if I don't see it, then you can interrupt.
Okay, perfect. Okay. All right. Okay, I think we can probably start now. Yeah, we have a few minutes. Okay, so welcome everyone. My name is Shankar, and along with me, I have Furuzan Farhani. In this session, we are excited to learn from our colleagues about the neural processes underlying development disorders in humans. The format for the session, as I was just saying, it's an interactive talk, which means the speakers have about 5 minutes introducing their topic, and following which we'll have about 10 minutes for question and answer. The way we are planning to bring people on screen to ask questions if you're interested. So you just use the raise hands button to let us know if you want to come online or on screen. Otherwise, you can also ask the questions in the Q&A and follow it up with on screen within braces so that we'll know that you want to come online. Since we are running a little bit late, I'm just going to keep the introductions very brief. So first up, we have Hajar Nakwa from University of Toronto. She'll be talking about the role of corticoamygdala network in neurodevelopmental disorders. Please take it away, Hajar.
Everyone can see my screen? Hi, everyone. I'm excited to share this project with you all. So this project investigates the relationship between behavioral dysregulation and the corticoamygdala network using multimodal neuroimaging. So just as an introduction as to what the behavioral dysregulation or domains that I'm interested in, there are many different domains of behavior, but these two domains that often encapsulate other domains are called externalizing and internalizing behaviors. Externalizing behaviors is the tendency to act in a way that harms others. So aggression, defiance, bullying, and oppositional behaviors. Internalizing behaviors is a tendency to act in a way that harms the self. So these are behaviors such as anxiety, depression, withdrawal, and somatic symptoms. Although these are two distinct domains, they are often highly correlated with one another, with an increase in one resulting in an increase in the other. Well, not resulting, perhaps an increase in one, increasing the tendency to increase other behavioral. other behaviors of another behavioral domain. And for a long time, these types of behaviors have been linked to variations or alterations in emotional regulation, which has led a lot of neuroscientists to think that the corticoamygdala network is a key network that is involved in regulating these different behaviors. So the corticoamygdala network, we can see it's kind of thought to be this green network over here. So the amygdala and the medial temporal lobe connecting to different frontal cortical areas. However, it can be a lot more than that. So, here we see the cingulum bundle. It's a white matter tract that connects the amygdala to so many other regions of the brain. And we are starting to see that there's a lot of interconnection with the different areas of the cortex and the amygdala, which play an important role in these behaviors. So the question of interest is, there is some literature that suggests a relationship between the corticoamygdala network connectivity properties and externalizing and internalizing behaviors in typically developing children. And we were interested in whether we can actually see that relationship in a transdiagnostic sample. So in addition to typically developing children, we have children who have ASD, ADHD, and OCD. So the overall hypothesis is that greater externalizing or internalizing behavior will be related to reduced corticoamygdalar connectivity properties. To categorize behavior, we use a parent-report-child behavioral checklist, and to measure network properties, we use different properties that were extracted from T1 functional and diffusion-weighted imaging. So just to get more specific, from T1 weighted acquisitions, we use the corticoamygdalar structural covariance, we use seed-based functional connectivity, and fractional anisotropy and mean diffusivity of the unsnipped fasciculus and cingulum bundle, which are two important tracts in the emotional regulation pathway. As our kind of model that we actually used, we had brain index as the dependent variable, and the connectivity properties as the independent variable, and then we covariate for age, gender, scanner, and a couple of modality-specific covariates. So here it kind of shows the overall linear model of interest. So our results are not as interesting, but the discussion may be interesting. So we did not find a significant relationship between any of the corticoamygdalar connectivity properties and externalizing or internalizing behaviors. So with structural covariance, we did not find anything significant, nor did we find it in resting state functional connectivity. These are unthresholded T-maps, so while we do see red and blue regions, they're not actually significant. And similarly, we do not find any relationship with diffusion properties of white matter tracts and either externalizing or internalizing behaviors. So just as a quick discussion, we did not find a relationship, which doesn't mean that a relationship does not exist, but there may be other reasons as to why we didn't find it. So I don't think we have the sample size to really conclude that it's a true null result. But I think the results of this study actually have a lot of important implications for future studies as to how we do transdiagnostic or relationships in transdiagnostic samples. So detecting relationships in these samples may require multiple ways to probe behavior that move beyond parent reports. So we only assessed the CBCL, externalizing, internalizing behaviors. Perhaps next time we can do a task-based fMRI paradigm, which assesses different components of these behavioral domains that we're interested in. Another possibility is that there is this new emergence of a general or overall psychopathology factor, and that tends to capture the variance across different disorders. So that may be more useful to use in a transdiagnostic sample. And there have been a couple of very recent studies that have used that. And, one of the, although we did have a relatively large sample size, so about 350 participants, we may need larger samples to detect these brain behavior relationships that inherently have small effect sizes, particularly when we are dealing with a heterogeneous transdiagnostic sample. Thank you so much. I feel like that was 5 minutes for me, but you didn't give me the 1 minute warning.
No, you are perfect on time, so I didn't want to interrupt your flow. So that's the applause. I think this is on behalf of the attendees. So please feel free to ask questions using the Q&A or the chat. As we are waiting for questions, I have a very an operational question. How easy or difficult is it to get these children to be in a scanner for so long to acquire these these recordings? I mean, in general, it's a little tricky with children, right?
Definitely. So I was very fortunate that I did not collect the data. We have collaborators who did that. But it is one of the hardest things because head motion was a huge problem in our data. We had to exclude many participants due to head motion in the scanner. But what a lot of them do is they kind of have mock scanners. They instruct the children, not to move, or they have different runs. So they may scan a child for 5 minutes, remove them from the scanner, give them a break, put them back in for 5 minutes, and then continue to do that between all the scans, and then concatenate all the images together.
Okay, got it. So even if other panelists have questions, please feel free to message or just unmute yourself and ask the questions. So we're still looking for questions, and I have another question. So in this, I forget which age range were included in this.
6 to 18. So children and youth, yeah.
Okay. Okay, so I think Shlomit has a question. Shlomit, why don't you unmute yourself and ask the question?
Yeah, I can ask it. I wasn't sure if. Okay, so yeah, my question is, what was the basic assumption that made you measure the connectivity and the structural differences in these networks?
That's a good question. So if you can share, see my screen now, I just didn't have time to put it before, but there have been a couple of studies in typically developing children that have found a relationship using either functional connectivity, different diffusion metrics, or structural covariance. And the question that we were really interested in is whether the relationships found in typically developing children can be translated to a shared transdiagnostic sample. So the measures of interest were really taken from these previous studies, which looks at typically developing children.
Okay, thank you. And what's next? I mean, what are you planning to do now?
Oh, so yeah, so this study, null results. That is always a little bit hard as a graduate student, but what was really interesting was that there have been a lot of papers that have been thinking about the best way to model brain behavior relationships. So the next step for us is to include more multivariate statistics. So we're looking at, using more PLS models, partial least squares, to actually do these analysis. They may tell us a lot more about the data as opposed to univariate analyses, and also using much larger data sets to see whether it may just be the fact that we were a little bit underpowered considering the heterogeneity of the population, but perhaps when we get to 2,000, 3,000 participants, we're able to leverage the sample size a little bit better. So those are going to be the following studies.
Thank you.
Thank you.
Yeah, just to get back on the age, do you include that as a factor in your model?
Yes.
Okay, got it.
Yeah.
So,
age, gender. Gender was collected, not sex. That's why I call it gender. And scanner, because there was a scanner upgrade, was included as covariates in all the models.
Okay, got it. Perfect. Do we have any more questions? So I think we can just move on to the next speaker. And thanks for the wonderful presentation, Heider. And yeah, applause again. So next we have Shlomit Becker from Albert Einstein College of Medicine, who will be talking about oscillatory processes in autism spectrum disorder. Take it away, Shlomit.
Thank you very much. So let me just, okay, can you see it now? Okay, so thanks for joining this Zoom. And I'm Shlomit Becker, and I'm working in the Cognitive Neurophysiology Lab in Albert Einstein College of Medicine in New York. And I'm working on predictive processing in autism, and I'd like to show you some data, but moreover, I would like us to discuss predictive processing in the broader sense of cognitive or neurodevelopmental disorders and how we can measure it in the brain. So I'll start with showing you the data and then the discussion. So information in the environment.
Just to interrupt you quickly, are you sharing? We are able to see the entire screen of your.
Sorry. It's on the. Sorry.
You can just like.
Just I have two screens.
Okay. Yeah.
So I always have a problem with it. Let me just reshare.
Yeah.
Yeah. Thanks for letting me know. Sorry, just a second. How is it now?
Yep, perfect.
Is it moving between the
Yes.
lines and everything? Okay. So information in the environment is usually normally highly predictive of upcoming events, and the ability to anticipate relevant events is advantageous for adaptive behavior. Temporal expectations are necessary for dynamic cognitive control. In autism, so autism is a neurodevelopmental condition that affects social communication. Individuals with autism, we know from the literature that they show deficits in using cues to generate expectation. And the phenomenology of ASD includes rigidity of routines, insistence on sameness, inflexibility, and altered use of prior knowledge. The rigidity is inability to mentally adapt to new demands or information. And a good example for how people with autism have high rigidity comes from visual illusions. So, for example, here we perceive this triangle, okay? Although it's not like explicitly outlined. So people with autism will not perceive this or will be less susceptible to this illusion as well as to other illusions. So the general aim of the following studies that I'm going to show you results from is to test possible neuronal mechanisms that are involved in altered prediction and rigidity. In the first one, we did it on children. We had 31 kids between the ages of 6 to 9 years old. We measured EEG from them, and we showed them the task that includes 4 consecutive visual figures, followed by an auditory cue to which they responded. All these visual and auditory stimuli were equally spaced with 650 milliseconds between them. So while there was no difference in the evoked sensory response, not in the visual and not in the auditory, between the typically developed and the ASD kids and the autism kids, there was a significant difference in the anticipatory activity. So if you can see here, there is a nicely gradated pattern that starts from the first, second, third, and fourth. So there were 4, right? 4 consecutive stimuli in the pattern. So with each one, the signal was lower. And this is a clear indication for contingent negative variation, or CNV, which is an indication for anticipatory processing. This indication was absent from the children with ASD. Then we wanted to know how entrained they are, or how entrained their slow oscillations are to this rhythm of presentation. So we just low-pass filtered those signals, as you can see here. So black is the TD and the pink is the ASD. And we can see here that the ASD are not as aligned to the rhythm of the stimuli as the TDs. We can see it also here in phase concentration. So kids with autism do not track normally these stimuli. Eventually, again from the same study on children, we can see that they had low intertrial phase coherence. So just to illustrate to you what phase coherence is, if we plot one on top of the other, their individual trials, we can see that, for example, here on the left, for this TD participant, they are very aligned. The phases are very aligned. For this ASD kid, it's not well aligned. So anyway, there was a significant difference of the ITPC between these two populations. And what's more interesting is that those mostly significant areas were prior to the time here in the dashed white lines, time of stimulus presentation. So this was the next indication that they had lower or altered predictive processing. We've just received very similar results from similar.
Amit, sorry to interrupt you. I think we are running out of time.
Okay, so just the last slide.
Yeah.
From adults, we saw the same very similar pattern. So higher ITPC for the TD and lower ITPC for the ASD. And just one very small thing that we find very compelling is that in a jitter paradigm, so that the times of the stimuli is not fixed, it is jitter in a small window. We see that while the TDs in black have a tendency to follow this rhythm, even though it is jitter, those ASD people do not. So we interpret it as they are more rigid. I'll skip this and I'll get to this. The point of discussion is, for me, I mean, unless you have other questions, is impaired prediction, flexibility, unique feature of autism, and how specific are impaired oscillatory activity in general or for autism. So, yeah.
All right. Thank you for the excellent talk. We are waiting for questions from the attendees. And in the meantime, so I'm going to ask you the same question as I asked Hajar. Like, how difficult is it to get these recordings from these, like in this particular population? Because we have tried, we thought of doing EEG experiments in my previous lab. I mean, it was not me, but a colleague of mine. But we ended up doing sleep recordings because that's the best possible way we can get. So, yeah. And EEG, as you know, I mean, it has its own set of artifacts that's related to movement and other things. So,
yeah, so definitely cleaning and cleaning the data is a major step, especially from kids and especially from kids with autism, which are, you know, not always stay still. But I can tell you, so first, yeah, it takes a lot of cleaning, and also we just had to discard some of the participants for noisy data. What's very interesting, and this comes from another set of data that we're now working on, they're very, very consistent in their pattern of activity. So EEG is very, very consistent even across time. So once you overcome this noise, this external noise, we find that the results are pretty strong and pretty reliable.
Hajar, why don't you just unmute yourself and ask the question?
You touched on this, so this is something I've been interested in. In neuroimaging, there are a lot of different ways that we assess quality of scans. And I wonder for EEG, like, is there any quality control procedure to ensure that certain recordings are useful, or are they just kind of discarded if they have any bit of noise?
So let me just repeat your question to say that I understood it correctly. You're asking if we have any way to control that the EEG, so that, for example, they were looking at the screen and stuff like that?
Or just assessments of the quality of the EEG, like what is a process? Because like you said, there is noisy data, but if that, you know, noisy data is all thrown out, or are there procedures that can help with self?
Yeah, so we have each data set is going through a pipeline of cleaning, and it's based both on trial by trial and also on channel. So we interpolated bed channels and we replaced them with an average of the neighboring channels, for example. This is one way to do it. We throw trials with more than X bed channels. We discard trials with, I don't know, like voltage that cross some threshold. There is like a set of criteria to decide what are, what bed data is. And it's slightly different from kids to adults because they have an inherent, like a higher baseline noise.
There's one question in the chat box.
Yeah. So we have a question from Emma, who doesn't want to come on screen. That's fine. Hi, Shlomit. Thanks for your talk. She says, we know from other tasks that kids with autism can and do make predictions. Example, Kathy Manning's work. So this is interesting to hear. I'm curious if you integrate the hierarchical nature of predictive processing into any of the tasks to look at, and not only look at simple predictions, but prediction errors and precision rating.
I'm reading the question, just a second. Errors and precision rating. Not yet. That's the simple answer. So I can just say that predictive processing might not be the perfect title or name for this kind of project, of this project that I showed you. I think that the more appropriate name would be predictive timing, because we're not dealing with content of predictions, which is some other projects that are being done in our lab. But I'm mostly interested in whether they can predict the timing of events. So there are like different models and different paradigms that we do to measure this predictive timing.
So there's one more question asking about, in addition to ITC, have you looked at phase synchronization between the presented stimuli and EEG response, like people do in auditory research? I wonder if something like this is possible for visual stimuli.
So we do have an auditory module for the last study that I showed you on adults. I'm not sure I understand your, if Arujo, if you can, I don't know, like explain what you mean by it.
Do you want to come on screen?
I just show you phase concentration in this data and also ITPC, but maybe you can explain what you mean.
Do you want to come on screen? If you just let us know in chat, we can probably bring you on screen. Oh, he's just writing it down. Okay.
If I understand correctly, what he means is if we look at the phase synchronization between the presented stimuli and the response, and not only between or within, the response itself, that is, I guess, what he means. So we did the phase concentration, which kind of aligns or measure the alignment of the EEG to the timing of presentation. That is the most closer to what you suggest here, but yeah, I'll definitely look it up.
So waiting for, we have one more minute. I have a quick question about the comparison that you showed in the evoke potentials. It, like, after each stimuli, it seemed there was a negativity, or was it actually, it started high and started going down, right? So
You're talking about this?
Yes, this one, yeah.
Yeah.
But for the TD group, for the ASD group, it seems it's at the same level as almost the fourth, right? There is not that initial increase or like the higher positivity. Do you have any idea why?
Yeah, I agree with you. So first, I just want to say that, that the waveform or how it looks really depends on the place that you measure it from. So and also the electrode that you reference, right? The other channels too. So this could be different. And yes, you're right that the whole waveform is different for the ASD than the TD, and they start off with just a lower voltage. Yeah, I agree with you. So, but overall, for example, here in the TD, we found a significant difference between the first and the fourth responses, and here we didn't find any responses like the same.
Got it.
You're right that the baseline response to the first stimulus is also in a different voltage.
Okay, got it. Okay, thanks again. So we can probably we'll bring up the final speaker for the session. We have Kaya Ding from Southeast University. She'll be talking about prefrontal activation in left behind children. The floor is yours.
Oh, sorry, just give me a second. I'm going to share screen first. Screen 2. Can you see that?
Yes, perfect.
Okay, great. So my topic is kind of like left behind children. The topic is left behind children a real reason for the children's social cognition defects. I'm going to bring fMRI study on the effect of the social interaction on the left behind children. Let's have a look at the graphical abstract. We could see from this figure, we have 3 groups of preschoolers, and we access their social interaction using joint attention task by fMRI. And the results show that the brain activations in joint attention with the stranger was higher than joint attention with the teacher. Another result shows, because we have 3 groups of preschoolers, another result shows partially left behind children involved higher brain activations than other 2 groups in the stranger IGA task. So I'm going to first I'm going to introduce my objectives of the study. The first one is investigate the activation difference of joint attention type and the familiarity of the interaction partner. So here we divide into 4 conditions, as we can see later in the process of the joint attention. Second is evaluate the activation difference between 3 groups of preschoolers. And here we divided into three groups of preschoolers. First is completely left behind children. It means whose both of their parents leaves for work, and the child lives in the rural area with their grandparents or with their relatives. And partially left behind children, whose only one parent leaves for work and another parent leaves for take care of children, non-left behind children is a child who live with their both parents in the rural area of China. The last one is assess the correlations between joint attention and other social cognitive abilities like cognitive flexibility and facial expression recognition. So I'm going to give the left behind children's definition here. in case you don't know that, a child considered left behind if he or she between age 0 to 17 years old and lives without their mother or father or both of their parents due to migration. So here is the definition, and I'm going to introduce joint notation here. In our research, we divided into two types of joint notation. It's initiating joint notation and response to joint notation. If you don't understand, there's really simple. I can explain later. If so here is the math. We conduct three kind of methods. First is scales. We use PPVT, the Peabody language, to Peabody to check their language skills, and the ribbon to check their intelligence. And we use tablet-based tasks, a kind of like iPad. We can point right or left, and then we record their accuracy and react time. The third one, we use fNIRS. We develop the joint notation task in fNIRS recording. Here's pictures to show. Figure 1 shows joint notation task schematic. Figure B shows an example of a child wears NIRS cap on. Figure C demonstrates an example of grant average WIMPs. Figure D shows that fNIRS channels. Yeah. The results, here we can see the behavior results. We could see the PPVT. Partially left behind children is higher than other group. And we could see the accuracy of the CF, I mean cognitive flexibility, and accuracy of FE is accuracy of facial express recognition. There's no difference between accuracy of FE, but there is difference between PPVT and accuracy of CF. Yeah, we can just go here. We could see the difference in NIRS recording. So in the stranger task was higher than in the teacher task. The brain activations when children interact with stranger was higher than they interact with teacher. And we could say in the three group of children, the partially left behind children, the activation in partially left behind children is higher than completely left behind children and non-left behind children. Here is the results in words. So I'm going to give the conclusion. Partially left behind children invoked higher neural activation than completely left behind children and non-left behind children in the stranger IGA task. Initiating joint notation with the stranger demonstrate higher activation than with the teacher. So here we provide a new slide to balance parent-child separation and family economic in rural China. So because in tradition we will think both of their parents lives for work in urban area was good because they can improve their family economic. But now we give the new slide, like maybe one parent leaves for work, one parent stay for home for take care of their child will be better for children's development. Thank you.
Thank you for the wonderful talk. So as we're waiting for questions from attendees, I have a quick question about were there any differences between the non-left behind children and the completely left behind children? Or
actually we can see from the behavior actually you can see from the behavior, actually the non-left behind children is kind of like a hair a little bit. No. Left behind children is kind of hair, but they don't have any difference. We can just say the accuracy of facial expression. You can say non-left behind children is hair, hair a little bit than left behind children, as well as accuracy of the cognitive flexibility. But they don't have like a significant difference.
Okay, so that's interesting. So
yeah, that's really interesting because I can explain, I think, because you can see from here we only have like a 26 completely left behind children. But we have like a 36 non-left behind children, and the data we collect is in the rural village of China. I think that will be a very small sample. That will be a reason.
Okay, got it. And I'm not very familiar with FNIRS in general. So, like, which regions would these be? Like the way you measured the frontal regions, are these like lateral prefrontal cortex or like medial, or where do you think these activations are being picked up from?
We just like we just test the prefrontal cortex.
Okay.
Yeah, where is that? It is here, just like here.
Okay, perfect, got it. Yeah.
Because our nearest was like a sports one. It's very easy to bring to the area to get experiment. So.
Okay, really interesting. And I'm sorry if I'm asking too many questions. If the other panelists have questions, please chip in. But I'm curious about what you think, why the activation for stranger is more than for a teacher. Is that a good thing, or do you think that's... I'm trying to understand like what we infer from these activations. Is having a higher activation for stranger, is that mean it's good for behavior, or is it bad for behavior?
We met that problem because there is lots literacy and proved evidence that children would be have more brain activation when they interact with like their mother. I mean the really familiar people. But here we found that's totally different. And yeah, I think is that because I'm the person who is going to that place to doing the experiment. I found they are very they have courage for me. They just want to play with me, you know.
Okay.
When I go into their class, when I go into their room, just like when I go into that room and they just want me stay there.
Okay.
I think that was the reason, because in my essay I give the explanation of that because maybe they are just stay in a small village and they couldn't see people from other place. They was like a curiosity with the stranger.
Okay.
Because the teacher is their key worker in their room, so they're really familiar with their teacher.
Okay.
So that's the reason. Yeah, that's the reason. Maybe they first say, "Oh, there's a stranger here." I'm really like exciting like this. So.
Yeah. Okay. Nice. Cool.
Can I ask a question?
Yes.
I'm not familiar what is the living situation in these groups. Is it possible that the single parent kid only have experience of living with another person who is like the mom or the dad, but in other cases the baby has the experience of living with more than one person, who's going to be either both parents or other people in the care facility, so they have more visual stimuli from like other people compared to a person who has only one parent and probably more like isolated compared to others.
Yeah, that's a good question. And because this research we are doing is according to a program is China Africa left behind children. So the villages they have like a as you can see, I got just 81 children, like half of them, more than half of them are left behind children. So we will conclude that situation you said. Maybe next time we can compare the children who is from maybe city area or who is from like living with like both their parents and live with both their parents and both with grandparents as well. Yeah, that's a good question, because in China there's a common issue of left behind children, but there is another common issue. It's like one child live with like 4 people. I mean, both parents, both grandparents. Yeah, that's a good question. Thank you.
All right. So I think before we get kicked off Zoom, I just want to thank the panelists again. And it was a great session. So I learned a lot of new things. And thank you. All right.
Thank you.
Thank you.
Thank you very much.
Bye.
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 Thank you so much for the introduction. Good afternoon, everyone. If you're in Pacific time zone and anywhere else, good time. My talk is, as Manu said, is about the properties of macroscopic dynamics in the brain that can make things either be linear to some extent or at least look linear. And this is very important because if you've done any kind of computational modeling, you've been to that trade-off at some point that you have to choose between accurate models and simple models, right? That's the trade-off that we always have. And there are different axes for that trade-off, right? We can choose linear models or nonlinear models. We can choose small models, for example, zoom into the brain or take the entire brain, so on and so forth. And typically, it is the case that the most accurate model that you can get ends up being very complicated. And you have to sacrifice your accuracy for a little bit of simplicity and tractability. And then you're always looking for that sweet spot of what is the right model for my task, how much complexity I should bring in, how much should I leave out. And this work is specifically about the axis of linearity versus nonlinearity. So, we know from Hodgkin-Huxley at least, or maybe earlier, that at the micro scale for individual neurons, the brain is greatly nonlinear. These are the Hodgkin-Huxley equations. There's hardly any linear term in there. There are tons of nonlinear terms, fractions and exponentials and all kind of things, products. And this is great because it's data-driven. It's not just based on some assumptions about the brain. It's based on actual data from the squid giant axon. But also we know that from a functional perspective, nonlinearity is essential for a lot of the things that the brain does. A linear system cannot spike, right? You need nonlinearity and the positive feedback and the state-dependent feedback of, for example, the Hodgkin-Huxley equations for spiking, for excitable behavior, for limit cycles, for chaos, for bistability, for a lot of things that the brain does. You need nonlinearity, and so there is no doubt that at the micro level, the brain is nonlinear. But the question is, when you zoom out and you come to the macro scale, now you have 86 billion or so
nonlinear machines that are connected together, plus glia and a whole lot of other things. And at a quick look, it seems like you will have a massively nonlinear system. It's just 86 billion nonlinear machines just coupled together. It just makes the nonlinearity scale up like crazy. And so that has essentially resulted in an assumption or presumption in a lot of computational work in neuroscience that if you want to have models, dynamical system models specifically, of the brain, it has to be nonlinear if it wants to be accurate. So linear models might at most give you, even if they do, they might at most give you a first order approximation, or not even that. So they're not accurate for sure. That is the assumption. And so this work was trying to answer the question of is that assumption actually true? And so specifically, I'm going to be talking about computational models of this form. So ordinary differential equations of resting state dynamics. So in the most general form, I will have some ODE that is describing the state equation of the brain. The dimension of the state could be arbitrary, and this is in principle or in general latent to the brain. So this is not necessarily what we can measure. And then we are measuring some kind of neuroimaging time series, which is the Y. In this case, we're going to be using fMRI time series and intracortical IEG or ECOG time series. And then there's some process noise and some measurement noise. A lot of models used in computational neuroscience fall in that category. Neural mass models are probably the most famous. All kind of recurrent neural networks fall into this category. And the basic question that we're trying to answer is, what is the best model for a time series that we measure? Notice that we don't have an input signal because it's resting state dynamics. It's not task dynamics, and that's important. So it's resting state is only driven by noise, by assumption. And then we want to see what is the best model that fits the data. We essentially went through a whole bunch of models that we could reasonably assume that they're relevant. We took so first of all, we discretized what I showed you in previous slide was continuous time. We discretized it because we have discrete samples of data. And we just took a very simple linear model, a whole bunch of other regressive models, different variations that I go dive into the depth of all the variations of autoregressive models that we checked. For the fMRI data, we went all the way to a model that includes the hemodynamic response function and learns the hemodynamic response function as well as the color of the noise and a whole bunch of complexities. And finally, a very standard state space model with latent states learned through the famous subspace myth. This is the bunch of linear models that we tried, and then a whole bunch of nonlinear models, including neural mass models with a sigmoidal nonlinearity over here, with and without the hemodynamic response function, deep neural networks, ReLU, deep neural networks with autoregressive lags, the manifold-based models that are locally linear based on the idea of local linear embedding in machine learning, that any manifold is locally linear, so you can essentially decompose nonlinear systems into infinitely many linear ones. And finally, the minimum mean squared error models that are optimal nonlinear models. They're just expected conditional expectations of what you want to predict. And they're optimal, so they give you a kind of upper bound on how well any nonlinear model can work, but they're very notorious to compute, so we cannot estimate them for the full model. We can estimate them either on a pairwise basis, so predicting each channel from any other channel, or on a scalar basis, which is predicting each channel on its own path. And finally, this is important, I always will put in the comparison list a so-called zero model, which is just a random walk. So it's essentially y dot equal to 0 plus noise. And this is important because we need an absolute basis for comparison. We don't want to just compare models with each other. We want to see if the models are any useful, right? Are they going to predict my data better than a random walk? So long story short, this is how they compare on the most important metric that we had, which was R squared. So this is out of sample cross-validated R squared for 700 subjects of the Human Connectome project, all of their resting state scans. The R squared that I'm showing here is just 1 minus normalized error. So y hat is just one step ahead prediction error of the model. for each of the models. Yi is just the signal, and you compare that with a predictor that always predicts at the average. So this is 1 minus a positive quantity, so it's always less than 1. If it's 1, it means that your model is perfect out of sample. If it's less than 1, your model degrades, so the higher the better. And it could be negative, right? Don't get confused with the notation, which is historical. R squared, in this definition of R squared, could get negative if your model does worse than a constant predictor. It could have a negative R squared. In the plot, these are box plots of 116 brain regions, 700 subjects combined into one distribution. And the black is the 0 model, which is our baseline for comparison. All the green are linear models. All the yellow are nonlinear models. And this is how well they fit to the data. And in this case, for fMRI, we see that the vector autoregressive model, third order, does the best. And I have to say that for all the models, we tune all the parameters. For example, the number of autoregressive lags for a vector autoregressive model, or all of these models have their own parameters or hyperparameters, and we tune them using a stochastic gradient descent kind of algorithm. And then this is the comparison between the best model in each model family. We also compared them on 2 other grounds: the whiteness of residuals, so how much dynamics is left in the residuals that is not captured by the model, and just the time, the CPU time that it takes to run the entire thing. And on all grounds, linear models do better, and specifically the autoregressive models do better on R squared and on residual whiteness. This is for fMRI. We had almost the same result for IEEG, except that now instead of a 3rd order autoregressive model, you have to go to a 100 order autoregressive model because your time scale is much finer. Your sampling frequency is much higher in IEEG. Notice that not every model is doing better than the 0 model, and all of them have an R squared more than 95 or so %. So the fact that your signal is very smooth gives you a very high R squared no matter what, but that doesn't mean that the model is a good model. You always have to compare it with a baseline and make sure that it is at least doing something better than chance. So the question at this point that we tried to answer was why, right? We have all the nonlinearity at the micro scale, but now we have time series completely blind to where the time series is coming from. We just take kind of a machine learning system identification perspective, and linear models just do better. And we try to explain that. Why could that be the case? And very quickly, I'm going to show you some simulation results that try to dissect that a little bit. So we kept 2 time series, X and Y, and coupled them through a nonlinear, for example, sigmoidal nonlinearity. And we just took an average over many, many instances of that. And what we observe is that as long as you even get to like 5 units averaged out, which is by far the case in the macroscopic brain signals, we average over many, many neurons to get into like 1 voxel or one electrode. As you start averaging out nonlinear units, you truly wipe out nonlinearity. This is averaging across the space. If you do the same thing across time, i.e. low-pass filtering, and there are a lot of processes natural in the brain that are inherently low-pass filter, the hemodynamic response function being the most notable. Again, as long as the cutoff frequency of your low-pass filter gets to about a 1/4 of the Nyquist frequency of your signal, you again almost completely lose your nonlinearity. Also, if you have no averaging at all, just add enough noise to your signal. As long as your signal to noise ratio gets to about 1, just the noise destroys all of your nonlinearity. And finally,
Irfan, 1 minute left.
Sure. Finally, if you have no averaging, no noise, just as you go from 1 dimension to like 100 dimensions, and if you keep the number of samples the same, you need infinitely many data points or exponentially many data points to capture nonlinearity with any learning technique, which we currently don't have. So in reality, we essentially have a combination of all 4, which is probably the case why we observe this superiority of linear models. So quick summary, for the context that I was talking about, resting state dynamics, macroscopic scale, linear models tend to be the winner on all grounds, and for the reasons of averaging across time and space, high observation noise, and limited data. And there are a lot of future work. I'm just highlighting one over here, which is, okay, we compared all the models, and linear models, autoregressive models were the best, but are they good enough? Right? Being the best does not mean that you're good enough for the task. For example, if you want to do control, are they good enough for model-based control? Which is something that we don't know, we're looking into. So thank you so much, and I'm open to discussion and questions.
So we have around 3 minutes for questions, and thank you so much, Irfan. That was wonderful. I can start off with a question. So how do you compare your work to the Hartman-Grobman theorem, for example? That is something that I tend to use in modeling linear models of behavior, where a nonlinear system near their hyperbolic equilibria appears to be linear. Is it the case that macroscopic brain dynamics are near equilibria? Could that be one of the reasons for linear dynamics?
I'm not sure about equilibria specifically, but it could very well be the case that this is because of the resting state nature in some specific region of its state space. And I was highlighting the resting state because if you have task data, for example, then it is presumably always possible to saturate one brain region with enough force, with enough input. So yeah, definitely. It could very well be the case that the resting state dynamics are not exaggerated enough to drive things into saturation or into crazy nonlinearities.
Thank you. And there was a question in the chat, which I'll read out, but reminder to all attendees that please ask the questions through the Q&A, not through the chat, so that other attendees can see it and also upvote your question. So the question was, what is the name of the notable low-pass filter that you mentioned?
Hemodynamic response function, probably the question is referring to, which is the neurovascular coupling phenomena or system, I can say, that transfers neural activity into the bolt signal, into what the fMRI captures, basically.
Are there any other questions from the audience? Seeing none, let's thank Irfan once again.
Thank you so much.
Thank you. Now we move on to Sivi Ashla. So Sivi's talk is titled Trajectory Prediction in a Biologically Inspired Network with Strong Inductive Bias. Oh, sorry, that's not. No. That was my mistake. His talk is titled Bayesian Networks, a Bayesian-like connectionist model displaying flexible learning and network-wide bursting. So Sivi is a co-founder and chief scientist at Optimizing Mind. And his most relevant paper about the subject discusses a symbolic neural network that uses self-inhibitory feed-forward feedback connections to enable recall in addition to recognition. So I'll hand it over to Sivi.
Thank you. Thank you for the introduction. Share screen. Okay. All right. So thank you. So the predominant recognition and learning models of the brain are either Bayesian network, which are actually the most brain-like because they have a logic type of reasoning through priming or preferences determined by priors, and also likelihoods, which is like the expectation of what the network is looking for. But the problem with this type of model is it's not connectionist. You must calculate distributions. And what happens is you cannot really build this model with theoretical neurons, and it's not scalable to large networks. And the other models are neural networks or feed-forward type neural networks, where basically you're solving the recognition problem by taking the input and multiplying it by the weights. And this is actually much more scalable, but there's no brain-like priming or expectation. And although it's considered connectionist, it's only semi-connectionist, because you can only model recognition through theoretical neurons, not necessarily learning. Because in order to do learning, you would have to deal with this gambling problem, which tend to be ignored, where you have to store all the examples, shuffle them, and present them one at a time. And neuroscientists tend not to do this. And I'll jump into a little bit more detail about what I mean by this gambling problem. So when you're looking at an environment, you might see something, let's say a bicycle, and you'll learn it. A car, you'll learn it. And another type of bicycle, you just learn it. And you learn the patterns just as they come by in life. And it's unsorted and it can come randomly. There's no rhyme or reason. But this is not true of the feed-forward models. They require carefully dealing the training data. And so a corresponding neural network timeline to learn the same thing, for the first pattern, you can just learn it. But if you see it with the second pattern, well, what you have to do is shuffle it, which is like dealing, like a card dealer, and then reshuffle it, present it to the network, reshuffle, present to the network until the network learns. And then you go through this process and you repeat. Now, the next pattern you want to learn, you have to start from the beginning, redeal, reshuffle, repeat, and so on and so forth. So all of this is not easy to implement with neurons, because neurons would have to learn each one of these patterns, store them, and then have a mechanism to pull them out and rehearse. And moreover, in a feed-forward network, the data must be carefully prepared and balanced. And what's even worse, as a neuroscientist, this is not implemented usually in neurons. So we see the models and we see the outcome, and we see the model during recognition, which looks very connectionist, but the learning is not connection, just by having to encode this sort of rehearsal. So I call this the gambling problem. And if you get anything from this talk, this is it. And on top of everything, neither of those two models display network-wide bursting. And what that is if you record neurons, and you, especially in a sensory system, and then you present some sort of a stimulus, then you'll see an increase of activation of a lot of neurons, and then they come back down and decrease. And you see this ubiquitously in many organisms and so on. But this is not inherent in these networks. You can model them, but then you'd have to put in integrators and things like that. It's basically not innate in the models. So I introduced this Bayesian model, which uses presynaptic regulatory feedback, and it's presynaptic. The output goes back to the input and inhibits it. And in this, the weights become the likelihood average. The priors are determined by the activation of the neurons, and so on. So I'm low on time, so I will kind of walk you through how this works. Basically, each input that projects to output neurons, well, those output neurons have to project back to those input neurons and inhibit them. That's part of this regulatory, inhibitory, homeostatic type of feedback. And that holds true in any input. So any other input that projects to outputs, well, they would have to feed back and then inhibit back into the neuron. And that's how this network is built, and you can intermesh these as much as you want. You just got to follow those rules where inhibition has to go back to the same inputs that it came from. And then you can draw this whole system much simpler. Instead of this, you can just draw them as feed-forward feedback connections back to the inputs. So how does this whole system work? What I will do is I'm going to compare feed-forward and this system. And so here I'm drawing just simple matrices with 2 inputs and 2 outputs. So there's going to be 2 output nodes, 2 input nodes. first, I will just show feed forward. So before I do that, I want to learn, let's say, that neuron YA is going to be active when X1 is 1. And neuron YB is going to be active when X1 and X2 are 1. And so these are actually the likelihood patterns. So in the Bayesian network, what you can do is just basically put that into the weights. Basically, this is your expectation. So you just put that into weights, and that's all that the model needs to learn. It then estimates the distribution. So the learning is simple. Now, if you wanted to do the same thing with feed-forward networks, you would have to iterate through these type of examples until you got the feed-forward weights, which are these. And now you would run that. So let's draw first for the example for the feed-forward. Well, feed-forward is just feed-forward weights, so you put them here, and then you can put in a test of one of these patterns, and you can look at the plot of the dynamics. And I'm not going to draw ones and zeros. I'll just do yellow for active, black inactive, and any sort of gray in the middle is some sort of value. I present this test as a stimulus. I do my y equal wx, and I get the right answer here. But this is the dynamics. They're not really dynamics. It's just you multiply it through. And then you put the other input you expect, and you get the same thing. Now let's do the Bayesian networks. You have the same outputs and the same inputs, but now you have this intermediate inhibition, which roughly is like an error. And so now let's draw the weights. So the weights are these likelihoods. So the only key here, I'm not going to draw 0 when it's 0. But if there's a weight going from input to output, there has to be a weight going from output to input and inhibiting back the same input. If there's 2 going up, there has to be 2 coming down, and that's it. That's your network. Now it's iterative during recognition, and this is a little counterintuitive. So we're doing the iterations during recognition to find the activations, not to learn the weights. The weights are the likelihood. So information goes up, forward, then down, back, forward, back, forward. And this is the dynamics that you get out of this network. So if you put in the test stimulus and it remains constant, initially both the inputs come up, and this is not what's expected. So then both outputs become active, and this in a sense causes a burst. Then information goes back down, starts inhibiting the inputs. Now this input has twice as much inhibition than this one, so it gets dialed down. And this is how the neurons become separated. So that input gets inhibited, but now when in the feed-forward pass, this neuron is more affected by this input than this neuron, so they start separating in activation. And so then the dynamics start separating out, and this process continues iteratively, and the dynamics continue separating out, until you get the same outputs that you would have gotten with the feed-forward networks, but you used the likelihood, and you didn't have to go through any of this rehearsal during learning, and learning becomes much simpler. And I'm going to show you a demonstration of that. What I'm going to do is take the iris data set because it's super simple. There are 4 types of inputs and 3 types of outputs, petals of a flower. So what I do is I train on data from only 2 of the flowers. I just designate 2 nodes, take all the data for those 2 flowers, and I train them, and then I test the network, and I get, and it's accurate. Then I designate a new node for flower 3, and then I train only on flower 3, no rehearsal whatsoever of anything that's been previously learned. And then I can test the whole network, and it's exactly the same as if I had randomized the inputs, presented all the flowers, rehearsed or not rehearsed. So this system allows much more flexible learning without that pesky rehearsal. And so this network shows this network-wide bursting. Here's another example. I trained a network on MNIST, and here I put in the dynamics of recognizing, I put in the digit 1, and I look at the dynamics of the circuit when it's going to recognize digit 1. And you see most of the digits burst for a little bit, and then they come back down, but then only the one that remains active is the one that ends up with the output. So in a big picture, in the brain, well, you don't know what's the mechanism of recognition, but it's connectionist, it's got priors, likelihoods, don't know what it's doing with the distributions, but it's pretty easy to learn and learn in a modular way where you can add a new idea at any point without having to rehearse old one. And it shows the network-wide bursting inherently. Now the Bayesian network, so what I've compared is the Bayesian network, the feed-forward network, and the Bayesian network. Now the Bayesian is not connectionist, and the neural network is half connectionist because you can't really build it up with neurons for the rehearsal very easily. And the Bayesian doesn't need that rehearsal, so it's more it's more connectionist. The Bayesian allows you priors. The neural network, the priors are embedded within the rehearsal of the training, so you don't really have access to them. They're there, but you don't have access. And the Bayesian network, you can slightly modify the activation of the neurons, and that's like putting a prior. And then likelihoods, Bayesian networks have them, neural networks don't, but the Bayesian does. And the way distributions are taken care of in the Bayesian network, you have to explicitly calculate them. In the neural network, they're set by the rehearsal. And in the Bayesian network, the distributions are estimated during recognition when you have the pattern that you want to recognize and you have the knowledge of all the other patterns that you've learned before. Then it takes those with the feed-forward feedback, estimates which ones should have the priority, which ones match the best. So it's neither explicitly calculated nor is it put into the training set. And so I've shown that it's easier to update. You don't have the scaling issues or the rehearsal. And you see this network-wide bursting. Wow, I'm on time. Any questions?
Thank you, Sui. That was great. I feel like, I mean, as I was going through your paper, I didn't quite get a handle on how Bayesian networks were a step better than feed-forward or feedback networks by themselves, but I think I get a really good idea. There are no questions from the attendees yet, but I did want to ask a specific question. In the network, how dense do the inhibitory connections need to be? Like, are they fully interconnected? Do they need to be?
No, they're as dense as the feed-forward connection because there's always symmetry. So if you've got, you know, a big tree going out, it has to come back in. I've done tests on scalability where I've basically made, you know, huge flat networks, more than you would in the feed-forward network. And it actually is able to deal with these huge networks better because it doesn't need those rehearsal process. So more inputs don't kind of inhibit all the adjusting of and sending back the error signals and so on.
And the other question I wanted to ask is, there are networks of the brain that are also hypothesized to consist of mostly inhibitory synapses, inhibitory recurrent synapses. And so do you have any idea, or are you planning to extend this to testing on actual neural networks?
Well, so my theory is that, so in this network, there's as many inhibitory connections as excitatory connections. right? It has to be. So I think that's already more realistic in some sense in the networks. And they're also top-down feedback. They're not lateral inhibition. They're not, you know, what you call recurrent in time. It's actually back to the same inputs, which is what you see in the brain. So, and you can't do kind of the feed-forward learning on that. Backprop won't work when you're inhibiting back to your own input, right? And so this is completely different.
You're saying that it's already a lot closer to what the brain actually does rather than what an artificial neural network.
Yeah, so the fact that you see this bursting, for example, where the network, when it's not expecting a signal and it gets something that it's not expecting, it kind of jumps up and back down. You don't see that in the feed-forward network. And I also thought this talk will be shorter. You also see some other phenomena which are more cognitive-like, which speaks more to the brain, like difficulty with similarity. So if you're trying to do a search between two very similar things, it's harder than if they're separate. And you see this in this network because if things are similar, the inhibition goes to the same place. And so it's harder to gather the activation. While if they're different, if they're more orthogonal, then they're more independent and faster and so on.
We have one question from the audience. I was wondering, how scalable is it in direction for a more complex cognitive task? This is from Kiribel Danieli.
Sure. Well, I can, let's see if I've got the slides for my, oh no, I don't. Of course I don't. Let me open up. Just give me a second. So I will show you my scalability tests. I believe it's this one. Let's see. Hopefully I got all of that.
If you cannot find it, if you can just mention it, that should be okay. To the audience, we are playing a little fast and loose with time because we don't seem to have the third speaker. That's why.
Yeah. All right. So here I've done basically I've taken just a crappy computer. And what I've done is I've generated random data and I increased the number of nodes and the number of input features. So this is the number of inputs, right? And so all I'm trying to do is see how scalable it is. And so, you know, the matrix size is just multiplying the number of nodes by the number of inputs. And I did this a while ago, but I was trying to emulate like what you can do on a cell phone, let's say. And so I compared support vector machines where I ran out of memory as soon as I got like 1,000 inputs and 500 outputs. While the fastest way I could figure out how to do a feed-forward learning, I ran out of memory at about 10,000 inputs with 2,000 outputs. But when I did it with this, I could go all the way up to 120 before it stopped. And so that's during learning. So I could do all the way up to 120. I couldn't do more than that because the random generator caused the running out of memory error. And then I compared during recognition, because during recognition, this method has to go back and forth and iterate. So I wanted to give an indication of how fast is this iteration. So here you have the computational cost per test for the feed-forward networks.
Oh, sorry, we're not, I don't think we're seeing your screen.
Oh no, so all of a sudden, oh, poop. Okay, sorry about that.
I didn't realize you were showing something.
Oh, yeah, I was. I have all this, all right, here, let me share my screen. Sorry about that. How do I share it? Okay, share my screen again. So this is what I was showing. Thank you for letting me know. Basically, I've set up a chart of the number of outputs and the number of inputs. And I just increased those as I go along, and the matrix size is just kind of multiplying these two together. And I want to see how scalable this is during learning. And I used a pretty crappy computer, but the point was to see, say, in a cell phone, how much you can do. And I did support vector machines, and it ran out of memory this is computing time. It ran out of memory, before I got to 500 on here, 500,000. Then I did the fastest feed forward learning I could, and it ran out of memory at 20 million here. And then I did the Bayesian network until 120,000. Beyond that, the random number generator ran out of. And then I compared with the computational costs when you're testing it. And so feed-forward networks they're really quick because you're just doing one multiplication, but they ran out of memory at 20 million here. So this is really fast, but it ran out of memory. With the Bayesian network, it just continued to increase linearly in the amount of time it took relative to the size, all the way to 120 to, here was about 2 seconds per test, right? So that's the measure, that's to answer the question about scalability.
Fascinating.
And then I tried to also compare this against k-nearest neighbors, and k-nearest neighbors take longer than the iterations that you do with the Bayesian network. I think I overkill answered that question, but
we had a little bit of time. So I wanted to ask the attendees if you have any questions for either of our speakers at this point. Going once, going twice. All right. If not, I wanted to thank Suvi for a wonderful talk, and thank you for staying over and answering more questions, and Irfan as well. And I especially wanted to thank Arash Ash, who was managing the back end of the Zoom room and talking to the attendees as well. And thank you everyone for attending the session. See you for the rest of NeuroMatch.
Thank you. Mute you. Awesome.
Perfect. So just wait like one more minute.
Yeah, he's here. Promote to panelist. Adam? Come on. Okay, well, go ahead and get started.
Sounds good. All right. So, yeah, welcome to session 27-22 in room 6. My name's Steve, and I'll be running the session along with Amit, who's going to be running this on the back end. And so we have 3 really exciting talks and a variety of different types of EEG research, including event-related potentials and seizure detection. And as a reminder, this is a traditional talk. So we'll have 12 minutes for each speaker to present, followed by 3 minutes of questions. And if you want to ask a question, you can use the Q&A panel. And also remember to upvote questions that you think are really interesting, because we'll ask questions in the order based on how many upvotes they have. You can also use the chat to share your thoughts and related information to the talk. So without any further ado, I'll first introduce our first speaker, which is Colin Hoy, and he's a PhD student at the University of California, Berkeley, in the lab of Robert Knight. And his talk is on multiple sequential prediction errors during reward processing in the human brain. So whenever you're ready, Colin.
Okay, great. Can you guys, let me hear actually. Looks good, and apparently you can see and hear me.
Yep.
Great, awesome. Yeah, thanks for showing up. So today I'll tell you about these multiple prediction errors that we're detecting with EEG in human subjects. And so, sorry, I'll try and kind of blow through things and give you guys the info, as much info as I can, and then maybe we can circle back to things that are poorly understood in the questions. But I wanted to start with the thank yous in case I do go over time. But obviously, my group, my PI, Bob Knight, Sheila Steiner is an undergrad that was instrumental to this work, and then the rest of the lab, as well as funding sources, Neuromatch, obviously, for hosting this amazing opportunity, Steve and Amit for doing the back end, and then whoever's out there for paying attention. And so I'll say that this is all stuff that's under revision. So there's a bioRxiv preprint, and there's a QR code at the end of the talk, along with a GitHub repo. And that's also going to be at the end of the talk. And so I'm happy to share slides or answer questions and such. So why do we care about multiple prediction errors and reward learning? Well, obviously reward learning is pretty critical for adaptive behavior, figuring out how to get reward and avoid punishment. And so reward prediction errors, and for those who aren't familiar, that's, usually have some expectation of reward. And then the reward prediction error is whatever the difference between the actual outcome and your reward prediction was. And we know this is a quantity that's central to the reward circuits of the brain, particularly dopaminergic circuits. And it's also something really important for machine learning algorithms and reinforcement learning, which are basically one of our best ways to explain complex behavior. And then lastly, these different learning signals, the prediction errors, are also linked to very different types of clinical symptoms. And so teasing apart all of these things will help us better understand behavior, and also it'll help us sort of better understand when behavior goes wrong in disorders. So, so the background on this is that the original finding of dopamine neurons encoding reward prediction errors was kind of a big way to link reward to the brain. And then in parallel, reinforcement learning also had great developments using these reward prediction errors, particularly for stimulus outcome expectations. So you get a stimulus, and then what do you expect the reward outcome to be? And then the difference between that expectation and, again, the outcome to get a reward prediction error is really foundational to algorithms like temporal difference learning that have been able to explain a lot of behavior. But it's also a hot topic recently because new tools like optogenetics have enabled us to really dissect these dopamine circuits in the midbrain and found new pathways that are separate from the classic reward prediction error pathways. They code for aversive and also non-valenced salient stimuli, so just surprising or alerting things in general. And also, reinforcement learning algorithms have really developed and incorporated a lot of these other prediction errors as well to model the world. So not just stimulus and outcome, but what is my action in between? And then what is the relationship between that action and the outcome? And so modeling all these different prediction errors, both the valence ones for reward and just the sort of surprise ones, like, I reached for the cup and I just missed, is, important for all these learning cases. And it's interesting because these are also related to some models of cognitive control that are a little closer to my work. So in parallel to the reinforcement learning in systems neuroscience, EEG has definitely been doing a lot of work on this as well. And so event-related potentials, ERPs, which Andrew's going to talk about it too, I think, are an important way to track different reward processes and learning in humans, right? Because it's non-invasive EEG. So one of the key players is the feedback-related negativity. This is an early, meaning about 200 milliseconds after you get some reward feedback, component that is central right here over the frontal midline in FCZ, which has been localized to the medial prefrontal cortex, which receives those dopaminergic reward signals. And it's localized to a theta frequency, which is 4 to 8 hertz. And really people have debated a lot about what's this mean? What is the FRN? And some key influential papers were arguing that it does track the reward prediction error and the valence of that reward signal. And there's a lot of evidence that really supports that story. But somewhere in the mid-2000s, people started to observe, hey, there's an FRN after surprising but positive outcomes. So it's not just that, a negative reward prediction is eliciting this. And they were arguing that, hey, this is actually maybe a salience thing. It's not valence, it's just overall surprise. And there's been a ton of papers back and forth for multiple reasons, but some we'll get into. And so it's a little bit unclear whether it's a valenced RPE or a salience. And this is important because, the FRN is predictive of some symptoms and onsets in depression and some substance misuse disorders and addiction. So we want to figure this out. But part of the problem is that the FRN is only one of the ERPs, and it's kind of mixed in with, say, the P300, which is a famous ERP. It's a little slower, so it occurs later in 300 to 400 milliseconds, and in a different part farther back in the brain, localized to posterior PZ sensors, and is slower in the delta range. And that's related to more surprise and model updating. So also surprise and prediction errors, but not quite clear how that relates to the FRN. But also has been observed to be abnormal in disease. So teasing these things apart is kind of the goal here. And just a disclaimer, if you're an ERP aficionado, there's a lot more subtleties you can pick into separating out the FRN and the P300. And there's a tidbit in that at the end. So here are what these ERPs look like. 0, here's a feedback, and then there's the FRN, this little dip at 200 milliseconds. And here's the P300, that positivity afterwards. And the problem is, teasing apart one while not being influenced by the other one is hard. And particularly given that a lot of the traditional analysis methods are just averaging over a window, right? So if I just take the mean in that, I'm probably affected by both. And then measuring some kind of distance between peaks can be problematic. And so a lot of the traditional methods run into trouble separating things out. So we're going to use a regression-based approach to get around that. And so first off, our task is a timing task. So basically there's a ring of lights and you see the light go around and you're supposed to push a button when it gets back to the bottom, right? And that's ideally pushing a button at 1 second. But if you push it anywhere in that gray bar, that target zone in the tolerance, you'll get reward. And so then at feedback, you either get a green ching if you win or a red bottom if you lose. And then we have a rare sort of neutral condition that doesn't tell you if you won or lost. And so importantly, there's surprise that's both positive and negative, depending on whether it's a really hard or easy trial. So we staircase the size of this tolerance interval in order to make it really hard or really easy. And that way we control whether you're expecting to win or lose and tease apart surprise from valence. And so that means that we get about 80% correct in everybody on easy blocks, 20% correct in hard blocks. And then we're going to model this behavior by looking at the individual trial outcomes. So on the x-axis here is the tolerance, the size of that bar, which is either really small or really big. And then the sort of 1s and 0s are the individual outcomes, wins and losses. And so for every one of those trials, we can do a logistic regression across the entire data set. And that'll give us the sigmoid curve that you can convert the tolerance on any given trial, which is that Q they see the whole time, to a probability of winning. And then that basically scales to the reward function to give us an expected value, right? And that's the first part of the reward prediction error. And so then we can contrast that with the actual outcome, the 1 or the 0, and get a reward prediction error, which is the tan plot here. And now importantly, we have single trial reward prediction errors, which we can correlate with the EEG features. And of course, we can take the absolute value of that to get a non-valence RPE magnitude, which would be like, how surprising is this reward? And based on our task design, we can also tease out a second set, a source of salience called the outcome probability, which is just how likely was this thing to appear. So there's 2 sources of salience and then the valenced reward prediction error. And what we're going to do is we're going to use that in a regression. So here's all our ERPs for all 6 outcomes in the easy, hard, win, loss, and neutral. You can see all of them have this dip for the FRN, the peak for the P3, and then some other stuff going on. And so instead of trying to average, we're going to go point by point across 50 to 500 milliseconds and do a multiple regression with all of our reward predictors and see which one corresponds to which feature. And if you look at the beta time series, so these are the model weights for those different regressors, you see that the tan RPE value, the valenced one, really peaks and has a strong effect right where that feedback-related negativity is, which is some good evidence that, oh, it's right there, nailed it on the head. And then the unvalenced RPE magnitude peaks with the P300 peak. And then that second source of salience that we separated out is actually this slower thing that's even later, kind of on the downslope of the P3. And so we've got 3 different effects corresponding to 3 different ERP features. And we can use this regression approach across space too. So if we look at a tiny little window of 50 milliseconds featured on the peaks of these different regressors, we can look at where on the topography they are, and that's here. So the columns here are 3 windows locked to the peaks of those beta regressors, again, for the 3 different regressors. And you can see that in the top panel, in the early FRN window, you have this loading of RPE value peaking on the front mid sensors where we know the FRN is maximal. Whereas at the P300 peak time, at 300 milliseconds, the RPE magnitude is in these posterior sensors where we know the P300 is maximal. And then we have this third salience outcome probability regressor that is even later and matches actually some frontal sensors, which is kind of an interesting effect that people haven't really teased out before. And then lastly, because again, we can apply the same regression approach across time frequency displays, we can see the frequency range of each of these effects. And again, you get RPE value in the theta time range, where we know the FRN is, RPE magnitude in the delta range, where we know the P3 is maximal, and then the probability is sort of in between, but slow and late. So we've separated these out in both time, space, and frequency by doing the behavioral modeling and the regression. And that really shifts us from trying to figure out the FRN to looking at this as an unfolding sequence of prediction errors. There's a valenced RPE with the FRN in this early mid-frontal theta. And then there's this non-valence salient sort of how surprising was this RPE, just the magnitude of it in the P300. And then by doing this modeling approach, we've teased apart even a third component that is just tracking the outcome probability. And so that's a kind of a key takeaway here, is that if you keep the high resolution in time, frequency, and space, then you can use this regression approach to separate out overlapping components, which is a really big challenge for the ERP field. So with that, I'll leave you with some future directions. So the aficionados of the FRN literature will know that there's some splitting between maybe the response for negative outcomes and punishment versus positive outcomes and reward. And so we're going to look at that because we've got a latency distinction where the FRN shifts around based on whether it's positive or negative. So we'll try and pick at that. But also we've got some intracranial data, and this is just a preliminary figure from 3 subjects showing you that the RPE value signal and the RPE magnitude signal actually don't overlap. If you look at the source of the FRN in this medial prefrontal region, they're all intermingled, but the different sites encode those different things separately. So no wonder they're mixed up on the scalp, they're mixed up in the brain. So those are the kind of future directions we'll pick at. So thanks. Anybody got questions?
Thanks so much, Colin. That was really fantastic. Yeah, if anyone has any questions, please put them in the Q&A. Yeah, I guess quickly while people are writing their questions, you talked about modeling, doing regression, which is fantastic for EEG. Really gives you a lot more than just averaging across conditions. Did you have to do anything to sort of deal with the fact that your sets were unbalanced? That the amount of I forget the word, like errors you had in the hard case was very different from the easy case.
Right. Yeah, so that's definitely a problem. So that's one place where this single trial approach helps us out. Rather than sort of averaging across conditions and then having, like one ERP per condition per subject, we use a mixed effects model where we throw all the single trials in, and then we have a random effects sort of intercept for the subject. And so I think that's really crucial to this as well. It's not just using multiple regression. I'd say there's a couple pieces. There's making sure that you really model the variables of interest. So don't necessarily just rely on an ANOVA contrast or, particularly in a 2x3 design like this, the interactions get hairy. Just model them directly from the behavior. And that's the first piece. The second piece would be use the multiple regression, throw all those things in the model and let them work out which one is really tracking what at this point, and apply that at every time point, every sensor at, the typical across the top of a window of interest, and then also, across the time frequency plot. But then, that's a lot of multiple comparisons to correct for. So the last piece is doing a single trial with a mixed effects model where you can throw in all your trials, try and keep all the variance of those, but then separate out the subject factor as a random intercept. And that gives you a lot more power than doing some like subject averaging and then a group level thing as well. And so, we have a pretty big data set. This actually, the nice part is I didn't mention, this replicates across 2 cohorts. There's an initial 15 subjects where I did all the piloting, and then a 17 subject sort of replication, and most of the effects replicate. The only things that don't replicate are those traditional measures of averaging in a window or the peak to peak measure, which says something, if those traditional measures weren't going to be able to tease it apart. So yeah, having sufficient power in the design for sort of single trial, modeling the behavior, doing a mixed effects with all the subjects in single trials, and then applying that across all your dimensions is a really good way to solve this sort of overlapping component problem.
Perfect. Yeah, good to know. Great. I think we'll move on now to our second speaker, who is Adam Lee. And so Adam is a PhD student at Johns Hopkins University in the lab of Sridevi Sarma. And his talk is on neural fragility of intracranial EEG networks towards an EEG fingerprint for the seizure onset zone. So whenever you're ready, Adam.
All right, cool. All right, thanks everyone. And yeah, thanks the moderators and especially NeuroMatch for providing this opportunity. So yeah, I'll get right into it. So I'm basically going to talk to you today about drug-resistant epilepsy. And these patients have very poor outcomes right now. Essentially, it boils down to 60 million plus people worldwide are affected by epilepsy. And then 30% of these don't respond to drug treatment. So the only treatment available at that point is surgery. However, only 18% of patients actually opt for treatment, whether it's surgery or stimulation. And that's primarily due to the fact that clinicians only accurately diagnose and treat the patients maybe around 50% of the time. And so this is, after taking essentially like a temporal lobectomy on your brain. And also hospitals lose money on each failure. So in epilepsy, there's this concept of the epileptogenic zone and seizure onset zone. And so epilepsy is characterized by unprovoked seizures. And this EZ, if you will, is defined as the minimum amount of cortex required to be removed to produce a seizure, or minimum amount of cortex required to produce a seizure. And so that definition I just gave you isn't actually rigorous. There's no way that any clinician or human, for that matter, unless you're God, you're able to know what the EZ is. And so really what clinicians strive to do is they try to identify potential seizure onset zones, and then they'll do a surgical excision. And they hope that this actually encompasses the actual EZ. And so the only true labels you ever have access to are the seizure outcomes, as well as the initial clinical diagnosis. And so I'll walk you through a standard clinical hypothesis generation. And so we'll start out with sort of a rough schematic of how epilepsy might be presented. And so in the red, you'll have, say, the ground truth EZ region that we'll never actually know. And the rest of the brain will be normal. And so there will be varying degrees of complexity ranging from a lesional epilepsy, which is something that you can identify on the MRI, to temporal lobe epilepsy, to extratemporal lobe epilepsy, to multifocal epilepsy, where seizures are originating from multiple regions of the brain. And clinicians will typically do ECOG or SEG implantations and look at various snapshots of recordings. And they'll try to visually identify various features of these EEG signals and basically try to localize where the seizure onset zone is occurring. They'll form a hypothesis, and then they'll perform subsequent surgery, and essentially an outcome will be measured based on whether or not they're seizure-free or not. So rather than analyzing EEG signals on a channel-by-channel basis, we take a different approach where we hypothesize that seizures manifest as a byproduct of unbalanced EEG networks. And I'll try to explain more rigorously what we mean there. So on the left-hand side, we have a balanced EEG network where, everything is oscillating, but nothing is straying away from baseline. So you can think of this as if you apply an impulse to a linear system, all its eigenvalues are within the unit disk. And then on the right-hand side, you have an unbalanced EEG network where you see that essentially after a certain period of time, large oscillations and large amplitude EEG signals occur. And we believe that this is due to an unbalanced EEG network where there's potentially imbalance of excitatory and inhibitory connections. And so the observation is that, well, we've seen this happen in rat and histology cells, essentially, where this can manifest due to loss of chandelier cells, where there's decreased damping in the network, or abnormal axonal sprouting of pyramidal cells, which leads to increased excitation in the network. And so we want to model this using computational methods. And so I'll bring you back to this slide where we don't actually have ground truth. We don't have access to the underlying EZ. And so instead, what we do is we take a look at what clinicians do, and we try to perform an analogous approach. So we have spatial temporal intracranial EEG maps, and clinicians use a concordance of various data modalities such as neuroimaging and non-invasive monitoring. And they work with a team to try to perform consensus to determine their confidence in their SOZ hypothesis, and then they perform surgery. So we take spatial temporal feature maps that are split by this hypothesized SOZ that the clinicians label. We construct a structured representation of these two signals over time. And we use an ensemble of decision trees, specific variant of random forest, to compute a probability of success for that seizure outcome. And so in this way, we can compare versus a ground truth label that we actually have, which is the outcome of the surgery. And here, note that if you have a really good feature representation of the underlying epileptic network, then you would think that this approach should provide you with the ability to be able to predict outcome very well. And so we take this approach and we apply a model that's described in the paper known as neural fragility. And I'm happy to answer questions on that at the end. But we basically construct these heat maps that are essentially one-to-one correspondences with the original intracranial EEG. And specifically, we take a sliding window, compute a linear system, or estimate a linear system. Then we estimate how fragile each column is, or each channel of the intracranial EEG network is to within the network. And we specifically compute how much energy it takes to destabilize the network, to basically shift its eigenvalues from within the unit disk to outside the unit disk. And here we present a heat map of an ECOG example, where the red electrodes on the left-hand side correspond to where the clinicians hypothesized as SOZ. Orange represents where they actually did the surgical excision. And on the right-hand side, we have this fragility metric, and the red electrodes correspond to these exact SOZ hypotheses. And then the color represents our fragility metric that we computed. Now, note this metric isn't computed using labels. It's not a machine learning classifier. It's just simply a hypothesis that we generated based on observations of epilepsy data. And if we look at the raw EEG itself, okay, well, we kind of see similar activity if we, I mean, I'm not an expert EEG reader, so I wouldn't know. And so I'll show you another example. So here's a successful laser ablation where I think the effect is even more pronounced here, where we have a specific ablation at a very small region on this depth implantation. And we see basically very high fragility of these, set of 3 channels. And even in the raw EEG, okay, I think there's some more oscillations, but here I'm kind of cherry picking for you to basically show you the best examples as possible, right? So then if we go to a failed example, what do we see? I think that's really kind of the harder question to ask. And so again, we have where the clinicians predicted or hypothesized as SOZ, and then we have everywhere else. And I think with a failed case, we would hopefully expect to see, as we see here, that there's a high degree of fragility or this feature value outside of where the clinicians annotated. And so this kind of drives us towards trying to quantify this effect across 91 subjects specifically. And so we perform an experiment over our data set that's listed in the paper, where we have 91 subjects and we perform a 10-fold nested cross-validation. And here we're reporting statistics on the test set. Now, granted, we're basically optimizing a classifier for every single feature that's listed on the x-axis here, right? And the x-axis just has a lot of features that are traditionally looked at in the epilepsy community, as well as the neuroscience community for that matter. And then, so we see that in terms of area under the curve and average precision, the neurofragility feature representation generally performs the best. And we can quantify this effect size as well as the statistical p-value. And we also still have the best performance. And so in terms of the p-values, it can be seen on the y-axis that, okay, pretty much everything is quote unquote significant. And I think that's a really good lesson to basically visualize other statistics as well, not just the p-value generally. But this is also good to know that it's an order of magnitude better in that respect. So the next thing we tried to look at was essentially looking at how these predictions are when they are stratified by clinical covariates. And so for the people that are unfamiliar with epilepsy, here on the x-axis on the left is the clinical complexity. So we would expect that these are, you know, from lesional to temporal to extratemporal to multifocal. It's harder and harder. So generally, you're more likely to fail, right? But we didn't optimize specifically on predicting clinical complexity. But we kind of see in terms of the success probability that was predicted that lesional and temporal lobe epilepsy patients will generally have a higher probability of success than these other ones. And so that's good to see. And then on the Engel class and the ILE class, these are just different stratifications of how much seizure was reduced post-surgery, where 6 and 4 are the worst, basically no change at all. And 1 is basically seizure-free, you can imagine. Now, so we optimized in this classification approach of 1 against 2, 3, and 4, but we see the trend from 2 to 4 decreasing as expected. And same with ILAE, where we essentially optimize 1 against the rest, but we still see a relative trend decreasing from 2 to 6, which is really great to see in the sense of, you know, matching against what clinicians are already using to label their seizure outcomes.
Hey, Adam, I just want to let you know that it's been a little bit past 12 minutes. So if you could wrap up in the next minute or so, that would be great. Okay,
yeah, sure. All right. Yeah, I have 12 minutes on here. Okay. Yeah, just saw it. So, all right, I'm just going to skip to the end. So what did I show you? That the EZ is a manifestation of a fragile network characterized by seizures. And one can compute this notion of fragility from a linear dynamical system network on every channel in a sample of intracranial EEG data. And neurofragility in this classification task that we pose outperforms traditional features. And in future work, we're exploring how it can be used interictally as well as on scalp EEG data. And yeah, I just want to thank funding and collaborators and LabMates and stuff. Yeah, thanks.
Awesome. Really fantastic talk. Thanks, Adam. So Colin has a question. Colin, do you want to just ask it?
Yeah, sure. I mean, one was a quick one. Is this computed on the full bandwidth signal or band limited?
Yeah, so it's band limited in the sense that so intracranial EEG, we generally get, for at least the 91 subjects we have, we basically had usually 1000 hertz sampling. And so I constrained the signal to be between 0.5 and 300 hertz. So it's just like trying to capture everything as well as possible without any DC shifts and as well as nothing crazy high frequency, basically.
Yes, I have a quick question on, so this fragility measure, is this, I assume this is sort of like between pairs of electrodes?
Yeah. So we actually have a pretty good example here, I guess. We have a quantitative two-node example, I guess, if you will, where you can imagine that just for simplicity's sake, we have an E and a excitatory and inhibitory node. And so going from left to right, we have, say, a linear system that's characterized as such. Now note that in our case, we're estimating the A, but here, okay, so we have the A. So say you don't apply this perturbation and estimate how much energy it takes to destabilize the network, then that perturbation norm is 0. And so if you apply an impulse response, it'll kind of be like, it'll act as a quote unquote balanced network, if you will, like the impulse response will decay to 0. Now, if you alter the connections on node I, or channel 1, say with a norm of square root of 5, you'll notice some difference in the impulse response. Now, if you modify just enough such that you shift the eigenvalues past essentially the stabilization point, then say that's square root of I, then you would have a seizure, right? And so this square root of I would be what we call the fragility of node I. And then you can imagine that we repeat this procedure across all channels. Hopefully that answered your question.
Yes, that does. Thank you. All right. Thanks again, Adam. I think we're going to move on now to our last speaker. This is Andrew Stewart, and he is a postdoc at the University of California, Davis in the lab of Steve Luck. And his talk is on data quality metrics to improve EEG/ERP research, the standardized measurement error SME. So whenever you're ready, Andrew.
Hi, everyone. Yeah, I'm glad we could see some cool talks of applications of EEG data. I'm working with Professor Steve Luck at UC Davis, who's done a lot of EEG bootcamp and coordination work of helping people run EEG experiments well. And we wanted to do big meta studies where we take not just data that we'd recorded in our lab, but also look at other recordings and combine them in a meaningful way to try and improve our confidence of the true measures that we were looking at. And we find it was kind of difficult to just put them together elegantly, because examining some of the data subjectively, we could see certainly there was data that was not brilliant and some data that was much easier to integrate. And I think it is the case that labs are going to differ. The exact protocols that people have, the hardware that we're using, and the way that we're training our RAs is going to lead to some amount of variance in the data quality. And especially since this is such tiny signal that we're examining. This is microvolts recorded across the skull, across the skin, which might be sweaty. There's so much environmental noise, even when we're in good conditions. And in order to look at more and more subtle experimental effects, we want reliable and repeatable investigations. We want things with high effect size, which needs both the true effect and good signal to noise ratio. There are many analysis procedures that can improve your signal to noise, perhaps different filtering settings, but necessarily this is going to increase analysis complexity. And so this is going to increase the experimenter degrees of freedom, and so likely greatly reduce the reproducibility. If we're doing things like collaborations or external validation or meta studies, these are things that we as scientists can do that will hopefully mitigate some of this. But these are costly in terms of coordination and in scientist time. And even these good practices, I think, could be better guided if we had well-established data quality metrics as something of a benchmark of our EEG data to say, are we recording this well or not so well? With Steve Luck, we were thinking, what is it that a good metric should do? What are the desirable and necessary properties? We want something that's going to be robust and consistent and sensitive to those things that are noise that's likely to be present. We want the metrics that we're producing to be interpretable and transparent, and hopefully very easily usable in people's typical analysis pipeline. But it is also necessary that they must be mathematically principled. I think it's the case that many people agree that that such a thing is an issue, but they think in their lab they're addressing it already. Perhaps if you already describe your own methods, and you know that your practice has been going okay so far, it feels like an internal replication. But I think I would say for a new data quality metric, just reporting your methods is not going to be sufficient to give people a sense of what your data quality is. I think it is good practice when plotting ERPs, as we've seen already today, to show the standard error across each time point. But this spread, just the standard error alone, it doesn't say whether your data quality is good or bad. It could be that your true effect does have some amount of spread. So I don't want things that just punish big effects. And I think it also makes sense that the metrics we use should not be blind to the experimental target. Something that is sufficient for an N for 100 might not be sufficient for examining a feedback-related negativity. It might be that we need different signal-to-noise ratios to establish these things well, and so different environmental situations. An example I think I can share with this is in the ERP core exemplar experiments that we have shared recently. We had a kind of reference P300 study in this, where along the time period of 300 to 500 milliseconds, something that's frequently done is measuring the mean amplitude within this window. And if you're examining a paper and it reports the P3 as this figure here, is the task of the reader and perhaps the reviewer of this paper to figure out, is this a good ERP? Is this a good study? Is this good data? And from this alone, this is difficult to figure out. But as I think you guys would agree, if you look at the single trials, you can get something of the representativeness of this ERP. There's something in the spread here where each single trial I've shown in a different color, where these 12 epochs average together to produce this ERP. The time period that you're interested in can go into this measure. What is it that we can do to have this put into one data quality metric? And what we're proposing is standardized measurement error, which we define here as taking the score in the time period where we have the standard deviation of this score of interest at this time period divided by the square root of the number of valid epochs here. So if there's an epoch that's getting rejected, this is not included in this. If there's another epoch that you can have in here that's typical of the same, then the SME is not going to be changing too much. We suggest that if we had extensive use of this and people were also reporting this, this would help researchers be better calibrated at assessing the data quality. But we do not take it to be that people have to manually calculate this. We have also written code that will calculate this on EG epochs, and we include this in our ERP lab toolbox. So if you're familiar with EG lab, there is the ERP lab toolbox that can be installed with this, where if you have a series of epochs and you run our ERP lab averager, now it will have this data quality quantification option, which is on by default. And custom parameters can be set, but by default, this will calculate this every 100 milliseconds relative to your stimulus onset. So for continuing the P300 example, it will show you for each bin. Take it here where we have 3 electrodes for the sake of simplicity. We have the frequent bin on top, the rare bin below. Each 100 millisecond period has the SME calculated within this. And here we can see, as might be expected, that the frequent bin has a lower SME than the rare bin, which has fewer trials and will have more change within it. The example I've pointed to so far here has been off the mean amplitude, which is something that many people report, but can have some issues. But I also want to stress that this SME can be calculated on other measures as well. So if somebody, I think as Colin mentioned in the first talk here, just measuring peaks can be quite problematic. But another measure that people often report is the half peak latency. So the time at which half the peak has been reached in the ERP. And measures like this, you can also get SME, but using bootstrapped resampling. So in a situation where you have frequent and rare trials, we would create many iterations and calculate the score on each of these iterations, and then examine the distribution of this to calculate the SME. And I also include some extra code in the new version of ERPLab to calculate this in 2 or 3 lines, where your average, where your epoch to EEG would be turned into perhaps 1,000 bootstrapped ERP sets. And we might take the measure of each of those and then save the SME of each of these, which could be reported in a paper. And I also want to stress that the code is available for this, and we have a preprint of this standardized measurement error paper available as well. And of course, the code and release and the binary release for ERPLab is available on GitHub as well. And thank you all. Thank you, Neuromatch.
Awesome. Yeah, fantastic talk, Andrew. So we have several questions. We'll see how quickly we can get to them. So in the Q&A, someone asked, if I understand correctly, SME is calculated at the individual subject level. How do you combine them for group statistics?
So these definitely can be combined together for group stats. We've found that in order to take SME and relate this to effect size and feature, it makes more sense to average together subjects using root mean squared combination. And at the point of doing the group averaging in ERPLab, this is automatically combined in RMS.
Awesome. So Colin has a question as well. So he's asking, have you or do you plan to compute SME for every panel of very common ERPs, like from published data sets? Because that would be useful.
Absolutely, yes. So we have, thankfully, the ERP core experiments, which we hope to be exemplars for many different commonly reported ERPs. So that has a P300, it has N400. I think it does not have feedback related negativity, but we got a few in there. And what we hope to do is release the information that we have of the values on this. We're not taking these as targets, as in, your experiment must be this good if you want to report it. But we want to have this as a kind of zoo of existing values, of typical values, rather than values to optimize towards.
Makes sense.
But what we hope to do is have an option in ERPLab where you could compare your current study to existing studies to see if your study is doing okay, or if there's things you need to do to improve.
Great. And then Adam asks, have you thought about integrating this into the BIDS specification for EEG data? Because it seems like it'd be useful for reproducible science.
That would be useful, yes. So we've had a few papers recently that we've tried to encourage using EEG bids as a default way for arranging your data. We've also used this for the ERP core experiments. We've ensured that this is specified in a BIDS compatible way. So far, we have reported the SME in the derivatives folder for BIDS and just kind of pointed people to that. But I think as they've done in fMRI, it might be useful to have explicit fields in BIDS that are at least encouraged for some reporting of the data quality in our shared studies. I think there might be such a thing for baseline values, but I think it would make sense to have more things in there as well. But so far, I've only put that in the derivatives folder for BIDS.
Great. Well, yeah, thank you all three of our speakers again. Fantastic talks, learned a lot. Really nice to see some nice breadth in EEG research. I also want to thank Amit for running the back end on this. Thank you to everyone who attended and asked questions. Yeah, bye.
Cool, thanks all, and thanks, Stephen.